what caused the disease, other than the fact that it was transmitted by DNA, which, as you think about it, puts the whole scientific paradigm completely upside down. That meant that when one had a good pedigree, by tracking the locus of the gene, finding the gene and understanding the gene, one could then generate a hypothesis about what caused and triggered the disease and how to treat it without initially knowing anything about it. So I thought then, and I still think, that this is magic. It's just pure magic. So we went down to the family reunion, and we set up a tent, as you can see here, a trailer. We stayed 2 days, and we examined and drew blood from virtually everybody in the family, which you see here. Now, at the top, there's a gentleman who first had the illness, who was, I think, irradiated for syringomyelia. But this was an extraordinary opportunity for us. We took the DNA samples that we had, and we studied them exhaustively, and were able to find a few markers, variants in the genome that were seen only with the disease and never without the disease. And of course, those mark the address of the gene. And when one delved into the genome at that area, it was possible to actually see which gene was mutated. I make it sound easy now. It took us a very long time. In 2001, we reported in Nature Genetics that we had the mutation in this family, which was a missense mutation in a subunit of a gene called serine palmitoyl transferase. And we did this jointly with a colleague in Sydney, Australia, who had another large family that he had studied. And so we published these back to back. So what is this all about? Well, I can describe that next. This is about molecules in mice. This shows the gene. It's a gene, serine palmitoyl transferase, long chain 1, which makes one of 3 long chain bases, so-called, that come together to form an enzyme. And in addition to these 3 protein subunits, there's another one, horrible name, mucoid-like protein, which inhibits LCB1. But taken together, this is a very important enzyme complex, which leads to the condensation of palmitoyl-CoA and serine. And I'll get to that in just a moment, but what we found in this patient, again, was 1 base pair change that led to a substitution of cysteine for tryptophan. So this is the condensation reaction that the enzyme promotes. You see palmitoyl-CoA, CoA here at the top, which is 16 carbons. You see serine, and after these are literally fused or condensed, you see with CO2 extruded, you have serine at the end here with hydroxyl group as shown. This is a compound known as sphingosine. Why is it important? Well, this happens to be the rate limiting step in the formation of ceramide, which in turn is very important in the genesis of sphingomyelin and complex gangliosides. So this is, of course, what one reads frantically to learn about once one has a gene mutation. And this seemed to be quite important and quite intriguing to us. This is a picture of the gene itself, which has 15 exons. And the mutations that we found were all literally in this region, exon 5, one of them was an exon 6. And a few years later, our colleagues at Queen Square in London found some mutations in exon 11. And taken together, this was a set of mutations that caused HSAN1, hereditary sensory and autonomic neuropathy type 1. It turns out, as a matter of fact, that S331F found in London was a very aggressive disease that started actually in early childhood, not just in adolescence. So that's kind of what we knew. Subsequently, the crystal structure of the serum palmitoyl transferase was deduced, and it was found that pyridoxal phosphate lays across an interface between SBT-LC1 and 2 at the active site, and that's critical in the way the condensation reaction occurs. And what we reasoned was that the C133W or our mutation C133Y would disrupt the active site. And indeed, when we looked in several family members at the enzyme activity, we found that it was indeed reduced, and unaffected members are in blue, affected members are in red, and it's reduced by about 75%. So that raises the possibility that one of the sources of this disease is loss of function. We went through a lot of efforts to make knockout mice, and what we found was that if we totally eliminated the gene, that the progeny were not viable, but that a 50% reduction in the gene and in the enzyme was asymptomatic. And that suggested, as did the fact that this is dominantly inherited, there must also be some component of the gain of function. So we made transgenic mice that had forced expression of high levels of the mutant enzyme, shown here as TGC133W, and we've subsequently been working on 133Y. And what we found was that when expressed in mice, this induced at about 1.5 years of age a neuropathy, which was indicated by things like slowness of withdrawal from a pain challenge, or latency to pull off of a rotor rod. If one then co-bred those mice with mice that overexpressed a very high level of the wild type gene, there was partial rescue. So that what emerged here was a mixed picture where there was clearly a toxic gain of function, but some component of loss of function as well. So what's going on? Well, we were very fortunate to collaborate with a brilliant biochemist in Vienna by the name of Thorsten Hornemann. And he had studied these chemicals, these compounds, and this reaction pattern for a long time. And he had found that when he looked essentially at this whole system, particularly in the proximal end of this reaction pattern, that he found in the patient's fibroblasts some abnormal metabolites that he had not seen before in humans, in human cells. And to cut to the chase, these turned out to be an abnormal condensation product in which the palmitoyl-CoA condensed not simply with serine, but also with alanine and glycine. So as you recall, we said that these two, at least introductory mutations, are right at the active site. And the idea is that they open up the active site, permitting access to the active site of alanine and glycine. And that when this occurs, they too condense with palmitoyl-CoA, now giving structures which have at the ends a slightly different appearance, of course, than when there is condensation with serine. And the one thing you notice is that the hydroxyl group is replaced essentially by a hydrogen-only group, depending on which particular species you're looking at. But there's no oxygen here. So these are called deoxysphingоids. And this first one is deoxysphingonine. This is the alanine condensate. So this gave us a sense that these might be critical in the evolution of this disease. And it was quite interesting that Penno, Hornemann, and others then took these compounds, and I don't have time to review this in detail, but co-cultured dorsal root ganglia with them. And indeed, these turn out to be toxic. So the idea was that compared to the normal pathway shown on the left, the mutant pathway led to essentially toxic substances which could not be converted to the higher sphingolipids and which accumulated. And this, of course, suggested the notion that this might be a biomarker for the disease. And remarkably enough, that's what it turned out to be. I would just draw your attention primarily to the bars on the right. So this is looking at both of these two different deoxysphingoid lipids. And what you can see is that they are markedly elevated in our mice in serum compared to the control mice. We're either wild type controls, wild type transgenics, or the double transgenics that have been rescued by crossing the mutant transgenic with the wild type transgenic. This is a very dramatic finding. It's what we've dreamt of in ALS. And then even more extraordinary was that when we looked at this in patients, we saw the same thing, which is to say that patients in their serum had high levels of deoxysphingolipids, clearly marking the disease. And that suggested that one might even be able to modulate the phenotype by playing with the law of mass action. You know, if you add more serine, you're going to drive more serine into the reaction. There should be less of the toxic metabolite. If you add alanine, there should be more of it. And indeed, in the mice, that's what we found when we put mice on a high alanine diet, they develop ulcerative lesions. You can see them here. And on high alanine, they have sustained high levels of the DSPs. When we put them on a high serine diet, that goes away. So this certainly looked like that was a clear part of this picture. But that then is kind of on the brink of some new phenotypes, which is quite interesting. So first of all, Thorsten Horneman said, gosh, this is a small fiber neuropathy. It's very rare. We always tell the NIH when we apply for money that this is the most common inherited small fiber neuropathy in humans, but in fact, it's incredibly rare. But it looks like diabetic neuropathy. And he asked the question, do you see deoxysphingolipid bases in diabetes? And indeed, when he looked, relatively small numbers, 50 controls, 50 diabetes patients, he did see elevations of the deoxysphingolipid. So and this was in the absence of mutations in the SPT LC1 gene, suggesting that the lessons learned from this incredibly rare disease might be relevant to the most common cause of a small fiber neuropathy. It's also the case then that this turned out to be relevant to an eye disease called macular telangiectasia. So one of the things my patient, Mr. LD, told me as he got later in life, and unfortunately, just before he succumbed to this disease, was that he had developed a macular eye disorder. And I didn't pay much attention to it, but as always, the patient was trying to tell us something important, and indeed he was too in his own way, and that was that macular degeneration turns out to be related to deoxysphingolipids. So this is a study that came out 2 years ago in the New England Journal of Medicine by Martin Friedlander and colleagues at the Scripps Institute. And what they did was examine a family that had HSA-N1, and what they found was that the affected members indeed had leakiness of the small blood vessels right around the macula. And this is a fluorescein angiogram that shows that, I think, pretty nicely. You can see that as compared to a control. So they then said, well, let's look further at this. They tried to look at other cases of HSA-N1, and they looked at some 10 cases in a row. And I think something like 90% of them had evidence of macular degeneration, if subclinically, but nonetheless evident. So then they did the experiment in which they said, let's take 125 patients who are known to have macular telangiectasia, not known to have HSA-N1, and who are unrelated, and compare them to 100 controls. And let's just see what they look like. And indeed, what they found was quite striking, and that is that in the patients with the macular degeneration, deoxysphingolipids were elevated as compared to the controls. Again, this is 124. This is, I think, 94 controls. And it's interesting that the macular degeneration people had lower levels of serine, again, suggesting that they were positioned less favorably in terms of the law of mass action for avoiding DSPs. And then within this group, the Friedlander group did something very interesting, which is they looked at the relationship between serine on the one hand and deoxysphingolipids on the other. And what you can see here is that as the serum level of serine goes up, the DSPs, the deoxysphingolipids, go down. And reciprocally, as the level of alanine goes up, the level of the DSPs goes up. So this embraces that notion of the law of mass action right here. There's not time to talk about it, but I will say quickly that the Friedlander group went on to use an in vitro model of an organoid retinal system to show that they could add DSPs and get really devastating loss of photoreceptors. And further, that there was a substance called fenofibrate that could reduce the level of the DSPs and benefit those cultures, a compound that can now be consideration for trial in these patients. But the next chapter is one that was reviewed brilliantly here by Payman Wahassel, who gave a neurobiology seminar here just a few weeks ago, in which it was shown that a different category of mutations in the SPT LC1 gene cause not HSA-N1, not DSP elevation, not macular telangiectasia, but ALS of a juvenile type. So he described how the Carsten Boneman lab at the NIH had studied 6 individuals with early childhood onset leg spasticity, toe walking, abnormal gait, with both corticospinal and lower motor neuron signs affecting all levels of the neuraxis, cranial, cervical, thoracic, lumbar musculature, without sensory or cognitive deficits. And in these individuals who met the diagnostic criteria for ALS, they saw no elevation of the deoxysphingolate bases, but they did see elevations in syngonine and circulating ceramide. And then they did exome screening, which led to the observation that these individuals had mutations also in the SPT-LC1 system. Just a quick view of this video. What you see here in this woman are tongue fasciculations, which are quite striking, with the rest of the face being strong, but the arms, of course, being devastatingly weak. You'll see a second patient in just a moment. Highly intelligent, cognitively intact individuals who have neck weakness, tongue fasciculations here as well, corticospinal signs as manifested by clonus at the ankles. You'll see the arm weakness here and clonus at the ankles. So clearly meeting the diagnostic criteria for ALS. And it turns out that these individuals, unlike those with HSAN1, have a different set of mutations, which are all in exon 2 of the same gene, which have nothing to do with DSP function, nothing to do with the prime function of this as a condensation enzyme, but rather have to do with the inhibition of this enzyme system by this ORMDL3 system. So this, in fact, the crystal structure of how this complex interacts with ORMDL3 is shown at the top, just came out several months ago. And it was possible to show that these mutations in these unfortunate individuals affected the interaction between LCB1 and ORMDL3. And we now know that there are some mutations in LCB2 that also affect the same interaction. So suddenly we have a completely different phenotype emerging from a different set of mutations in the same gene. What about therapies? I'll be brief. If you believe in the law of max action, you can ask the question, serine supplementation improve the neuropathy? Will alanine make it worse? So we took the set of the patients and we put them on low doses or high doses of serine for about a 14-week period. And within only 4 weeks, we were able to see an improvement in levels of the deoxysphingolate bases. We then did a study, and this was done largely by Florian Eichler, over 2 years, a long study of 16 patients treated with placebo or high dose serine. And then in the second year, all treated just with serine. And what we found was that it was possible to reduce the level of the deoxysphingolate lipids chronically in these patients using high dose oral serine, and that this led to some quantitative improvement in motor performance, and it was well tolerated. And I will just say that one of the amazing things about being at UMass was access to these extraordinarily brilliant people, such as Anastasia Kvarova, Jonathan Watts, who have made it possible to think about devising ASOs and siRNAs for suppression, either both alleles or allele selectively of SPT-LC1 and SPT-LC2. And those programs are now underway here. We're lucky to have in our lab Huiyu Yang, who's done a fantastic job pursuing this jointly with Jonathan Watts. So that's really the story. It's been fun over the years seeing how what we thought was an incredibly rare disease could in fact have relevance to some very common diseases, and also how the careful molecular analysis might lead to potential therapies. So I'm delighted to tell this story. I don't know if we have time for any questions, but that's where we stand.
Thank you so much, Dr. Brown. This is such an inspirational story for young people to start with, clinical observation and then have a nice collaboration with scientists, or the other way around, And I will definitely, we have a few more minutes, at least couple minutes for questions.
I have a question. Go ahead. Others go. So I had a question about the fluorescein. Are those actually the lipids that accumulated in the eye then? The accumulation of the lipids that are not metabolized? And do you see those in other tissues as well?
Yeah, no, what one sees there is the fluorescein leaking out of the blood vessels. So one gives a dose of fluorescein intravenously, and it circulates rather quickly up through the eye. And then one looks fluorometrically at the retina, and you see a blush as the material whooshes through the retinal circuitry. And what then happens is that when there's a leakiness, it leaks out and doesn't clear quickly.
Oh, so okay. I thought it was like lipofuscin or something, like an accumulation of the lipids. Okay, thank you.
No, it's a dye. It's a dye that it's used clinically.
Yeah, so when lipids aren't metabolized, they accumulate into like a lipofuscin. So that's, I thought there was a connection there. Okay, thank you.
Absolutely, absolutely.
And we have definitely more time for Dr. Brian Silver question.
Well, this is more of a general question. This is obviously a very specific mutation with what appears to be a very specific treatment. I just wondered in the general population, if people have observed people with dietary serine deficiencies, whether they have problems as well. This really raises a whole, question about sort of dietary influences on muscle function.
Huge. So in 125 people, these are people without mutations, they found the ratio of alanine to serine in the blood made a big impact on the level of these toxic materials. Brian, that's a super and a very important question. And the Friedlander group went on to show that they could take wild type mice and deplete them of serine, no mutation, just deplete them of serine, and the mice would develop DSPs in the blood, detectable. So what this clearly points to is yet another arena in which the balance of various nutritional components makes a big difference in terms of neuronal health.
And do we know if in the normal process of aging, whether that balance changes? Just thinking about sarcopenia and the influence of diet.
Yeah, that's another very interesting question. The answer is no. We have found some individuals with small fiber neuropathies who have mutations in the genes that make serine, wondering if there are other ways that system can be implicated. Needs to be looked at in aging. I don't think it has, Brian.
And we have the pleasure to have Dr. Riskin with us today, and he has a hand raised for a while for a question. So we'll do an extra time for him.
Thank you so much, Carolina. Bob, that was, of course, a fantastic talk. What a laboratory bench to bedside demonstration once again. I wanted to ask maybe an obvious question. What about the DSP levels in other small fiber neuropathies? I assume you've screened other people?
No. We've done a terrible job. We first of all, very few places have the assay, but we've been lucky with the proteomics core here to get the assay up and running. So we now can do that ourselves, but that's only recently. So no, we've not looked carefully enough, but clearly a good, and then the question is, if it is elevated, let's say in diabetic neuropathy, many questions. First of all, how do these DSPs get formed if there's not a mutation? Well, that might be the dietary balance thing. It might be something about dietary metabolism. Then the other question is, would some of these dietary interventions be beneficial in diabetic neuropathy? I mean, what I love is the idea that, again, a very, very, very rare disease has taken us to possible insights into the most common cause of small fiber painful neuropathy.
Incredible.
We really have to explore.
Yeah.
Thank you so much for all the questions and the great answer. And I think we thank you again, Dr. Brown, for giving us the keynote. We will move forward and give the scene to Janice Dominov, who is assistant professor in our department. And I will let her go with her presentation.
Hi, I'm Janice Dominov, and I'll be sharing some information on dysferlinopathies and our interest in developing new therapeutic treatments for this disease. I have no conflicts of interest to disclose. Dysferlinopathies belong to a group of diseases called limb-girdle muscular dystrophy. In the case of dysferlin, it's limb-girdle muscular dystrophy type 2B, or with a more updated recent nomenclature, 2R, to reflect that it's a recessively inherited disorder. There's at least 35 different proteins that are associated with various forms of limb-girdle muscular dystrophy. And they have similar features in that often presentation is in the proximal muscles of the hip and the shoulder muscles. With dysferlinopathy, there's a different presentation form called Miyoshi myopathy, in which the more distal muscles are affected first, but then there's a gradual progression of the disease, and eventually, regardless of the initial onset, the similar muscle groups are eventually affected. So it is the same disease, and why there's a different initial onset, it's not clear. As I said, it's a recessive disorder, so it takes two mutations to induce a disease phenotype. Among the limb-girdle muscular dystrophies, dysferlinopathies are among the more prevalent forms, with a prevalence rate of about 7.5 per million. So typically in neuromuscular clinics around the country, there may be about 2,000 cases that are being seen. The onset of the disease is teens to early adulthood, and up until that point, the patients are usually quite active. So it comes as a surprise that these athletic people then begin to exhibit neuromuscular disease progression. It's a slowly progressive muscle weakness, and its underlying cause involves muscle degeneration and inflammation. And it's the cause involves abnormal membrane repair and vesicle accumulation. So with progression over time, there's a replacement of healthy muscle tissue with a lot of fat tissue and fibrotic tissue, and typically patients become wheelchair bound. In terms of the structure of dysferlin, it's a large 237 kilodalton transmembrane protein, a member of the ferlin family of proteins that's involved in membrane fusion and repair. There are 7 C2 domains, which are calcium lipid binding domains, and there's other factors that can bind along the length of dysferlin as depicted in the figure. There's a large gene of 233 kilobases and a large transcript of 6.5 kb. There's 14 known human isoforms, but the main isoform in skeletal muscle is isoform 8, which is comprised of 55 exons that get spliced together. Dysferlin functions in membrane repair. So it's a membrane-bound protein, as shown by these little blue rectangles here, and is found both in the sarcolemma and also in membrane vesicles that typically reside underneath the sarcolemma. When there's muscle membrane damage, there's an influx of calcium, and being a calcium-sensitive molecule, this triggers the formation of complexes with other molecules such as annexin and MG53, and facilitates basically a patch formation, bringing the vesicles to the surface to patch up the hole in the membrane. So the role of dysferlin is in promoting efficient membrane repair. This process will happen in the absence of dysferlin. However, it'll happen at a much slower rate. And the thought is that over time, with slow repair of membranes, that eventually takes its toll on the muscle fibers and leads to the loss of muscle fiber function. In thinking about therapeutics to treat dysferlinopathy patients, these are exciting times with a number of exciting new innovative options for trying to address protein deficiencies. Among them, CRISPR-based editing methods and AAV viral delivery methods to try to restore protein function. We've decided to focus here on the antisense oligonucleotide-mediated altered splicing or exon skipping approach. And this stems from the great successes that have occurred recently in the world of Duchenne muscular dystrophy therapies and also spinal muscular atrophy therapies, among others. Highlighted here on this slide is just a list of some products that are now in clinical use to treat patients. And these products, or these antisense oligos, have been designed to target specific lesions within either the dystrophin molecule or the SMN2 gene to modulate the splicing patterns of these genes to restore at least partially functional protein to help patients. And so these offer great hope for patients with these diseases. So in the case of Duchenne Duchenne patients, these skip exons 51, 53, or 45 regions to maintain the expression of some dystrophin protein. And in the case of the SMN gene, SMN1 mutations cause the severe disease, and modulating splicing of SMN2, a similar gene, to force the inclusion of an exon 7 will stabilize that protein, and that can substitute for the absence of SMN1. So just to highlight, there's several different chemistries that are involved in this, and there's always the ongoing efforts to improve the chemistry of antisense oligonucleotides to improve the stability and the efficacy and the safety, which is always of primary concern. Dysferlin was actually discovered by Dr. Brown and his colleagues in the 1990s, and as such, he's had a long-term interest in dysferlinopathy patients and follows a number of them in the clinic. Among the families, it was one with two brothers who were affected by this disease. They had one known pathogenic allele, which was a stop codon in exon 32, but their second pathogenic allele had not been identified despite repeated attempts to sequence the exons to look for another pathogenic variant. So in this case, with the exon 32 mutation, there's the normal TGY is replaced by an AA, and that creates a TAA stop codon, and there is no further translation of the protein beyond exon 32. However, this lends itself to exon skipping strategies, because if we can force the splicing of exon 31 to 33 and bypass the stop codon in exon 32, we can maintain the reading frame of the protein and create a protein that may miss exon 32. We will lack an exon 32, so it won't be a normal protein, but it may restore some partial function of the protein. We've been able to do this. We've been able to design antisense oligonucleotides that target specific regions in exon 32, where normally splicing enhancer factors will typically bind to allow the exon 32 to be spliced in. And with the antisense oligos blocking the binding of those factors, it inhibits the process of exon 32 splicing. And when added to cells, we can see at the RNA level that we can promote the skipping of exon 32 in these cells. So shown down here is just on RNA blots, RT-PCR assays, where this is the normal splice form, including exon 32. And if we have buffer controls or scrambled oligo controls in these cells, this is the normal size. However, when we add antisense oligonucleotides, it promotes the skipping of exon 32 and the formation of this lower molecular weight RNA form that lacks exon 32. So we had the patient cells to study exon skipping in vitro, but we wanted to create a mouse model to be able to do in vivo studies. So for this, we used a CRISPR-based mutagenesis approach, and we introduced the TG to AA mutation in the mouse sequence. And as you can see, when we have homozygous mice with this AA mutation, there's absolutely no dysferlin protein made in these mice due to the stop codon. And this is using two different antibodies. So this provides a nice in vivo model to study exon skipping and possibly other therapeutics. Using muscle cells derived from these animals, we were able to show that we can use antisense oligos to force the skipping of exon 32 and the restoration of protein in these cells. This is shown at the RNA level by the appearance of the low molecular weight exon 32 skipped band in a dose-dependent fashion, and also in the dose-dependent expression of protein using two different antisense oligos. In this case, these are peptide conjugated PMO oligos that have been used on these cell cultures. So we're encouraged by the fact that this exon skipping can affect protein expression in these cells. We were a bit disappointed, however, when we tried to do the same thing in vivo by injecting these PPMOs into the exon 32 mutant mice. As shown here at the RNA level, we can see varying levels of exon skipping of the exon 32, evident by the lower molecular weight band in different muscles, but it is quite variable. And even going out to, this is a 3-week time point after injection, and then going out to 12 and 16 weeks, we can see evidence of exon skipping, but it is not complete, and it is a bit variable. And more disappointing is that we saw absolutely no protein being expressed, even going out to 16 and 20 weeks with injections in PPMO treatment. So either we're not inducing enough exon skipping to allow protein expression, or perhaps the protein that lacks exon 32 in the in vivo context has some stability issues. So at this point, we've not been able to see evidence of protein being expressed by exon skipping in this exon 32 model. But fortunately, while this work was ongoing, we identified another mutation in dysferlin that we think provides a much better target for our studies of antisense oligo-induced exon skipping. So in this case, we used the same patient cells that we've been using previously. Remember, these patients had one known mutation, pathogenic mutation in exon 32, but the other had not been identified despite a lot of exon sequencing to try to find it. We took the approach of sequencing the RNA from the cells of these patients, and in doing so, we identified an aberrant splice form in the RNA. In looking at this, what we found is there's a piece of sequence that is derived from the intronic sequence in intron 44 that is aberrantly included in the mature message in these patients. So we call this pseudo exon 44.1 as an aberrant splice form. And this actually encodes, this 177 base pair sequence encodes a 59 amino acid that gets embedded in the mature protein. This is caused by a single point mutation in the intron, so that's shown here. So here's the exon 44-45, and this is the intronic sequence. A single base pair mutation, a G to T mutation here, creates a new splice donor site that allows the aberrant inclusion or splicing of this pseudoexon 44 into the mature message. So rather than having the mature transcript looking like this in a normal and the mature protein looking like this, in the case of these patients, this single base pair change allows the missplicing, inclusion of the pseudoexon 44, and an inclusion of a peptide in the C2F domain of the protein, which disrupts its function. What's exciting is the pseudoexon 44 mutation provides an ideal target for exon skipping studies. We design antisense oligos to target the pseudoexon. We can block its inclusion in the message and restore normal splicing between 44 and 45 to make a normal message and normal protein. And we can do this as shown in vitro, looking at the RNA level here, where here's the patient cells that express the aberrant splice form, including the pseudoexon 44.1. When we add antisense oligos to these cultures, as opposed to scrambled or TE controls, we force the exclusion of the pseudoexon and the restoration of a normal transcript form, and hence these cells should produce normal protein. And looking at these same cells for protein expression using Western blots, we can see that antisense oligos induce the expression of the dysferlin protein in these cells. And here is just a quantitation of these blots. So with the success of our in vitro exon skipping studies, we wanted to create a mouse model to be able to study this in vivo. To do this, we used a CRISPR-based mutagenesis strategy in which we replaced the mouse sequence between exon 44 and 45 with the equivalent human sequence shown here. While the exon sequences between mouse and human are quite similar, the intronic region is quite different, so we wanted to create a good humanized target. We now have 4 founder mice that have this sequence replaced, the human sequence replaced in the mouse, so we're very excited that we'll be able to get these in vivo studies underway soon. In addition to the deep intronic mutation in intron 44 that leads to the aberrant inclusion of pseudoexon 44, we've been able to identify another deep intronic mutation in a different set of patient cells. These cells were sent to us by the Jane Foundation for patients that had one known allele, but their second pathogenic allele, again, had been unknown. We sequenced the RNA and again found an inclusion of an aberrant splicing pattern such that a piece of the DNA from within the intron is included in this mature RNA of these patients. This leads to a peptide that's being translated, but there's a stop codon here in this case, which truncates the protein. Again, as with the previous mutation I showed you, this is caused by a single base pair mutation deep within the intron that causes a new splice donor site to be formed and allows the inclusion of this pseudoexon 50.1 to now be included in the mature transcript. And when it's included in the mature transcript, again, there's a stop codon encoded here that truncates the protein, loss of transmembrane domain, and hence loss of functional protein. As with the pseudoexon 44 mutation, we've been able to develop antisense oligos to treat cells and show that we can use them to exclude the pseudoexon and restore normal transcripts and restore protein expression in these cells. So in summary, we've developed models for 3 different dysferlinopathy patient mutations, and these may be useful for developing exon skipping therapeutics and possibly other therapeutics. First of all, we have the partial restoration model that involves exon 32 stop codon mutation, where if we bypass that with exon skipping reagents, we will produce protein, although it will not be the normal protein and may have partial function. And then more excitingly is the full restoration models, the intron 44 and intron 50 mutations that encode the pseudoexon inclusions, which when we use reagents to bypass these will restore normal protein. So that'd be the ideal situation for testing these reagents and looking for efficacy in vivo and the outcomes at the phenotypic level. So we actually have made antisense oligos and used them to induce the dysferlin protein in vitro, and we're optimizing these reagents now, and we're very excited about moving them into our new in vivo models to test the efficacy of exon skipping to bypass the pseudoexon mutations. I'd like to acknowledge others involved in this work. Dr. Brown, of course, with his long-term interest in dysferlinopathy patients and ways to help them. Osgun Uyan, who was instrumental in the RNA sequencing work, and others in the lab that helped with various aspects of mouse work, and then other colleagues that supplied materials for this project.
Thank you so much for this wonderful presentation. Janice, we have a minute, but we can extend another 1 or 2 minutes, and we'll take that from the break. If there are any questions for Janice, please go ahead. I have, okay. Yes, Dr. Riskin.
So that was a wonderfully elegant talk, and I'm going to risk an incredibly dumb question. So in the context of dysfunctional dysferlin, and the cell has to use this alternative membrane repair mechanism, is that a much more energy requiring mechanism than when you have normal dysferlin? And if so, is there more metabolic stress on the cells? And if so, is that something that could be therapeutically approached?
Well, I wouldn't call that a dumb question at all, because it's very pertinent. And I would guess, my answer to this is I really don't know. I don't think it's really been explored, the actual energy requirements between different patients and in different systems. So I think it's a very good and valid question. My guess would be that the cells probably do have to work a bit harder in order to maintain their membrane integrity, since they don't have as rapid and efficient a mechanism for doing that. So the actual metabolism of the dysferlinopathy cells, perhaps there is some data on that, but I'm not aware of it off the top of my head, but it would be something worth looking at, I think, just to see overall if that contributes to the decline of the muscle, because muscle is very highly energy dependent, right? So that's a very good question.
Daniel has a question too.
Very nice talk, Janice. Wondering if you can say a word about the phenotypes in the mice. Surprised that they have some of them have no dysferlin expression, and just what do you see in the mice in terms of phenotypes?
Yeah, so the confounding thing, this happens very often, is between mouse models trying to emulate patient samples and patient disease. The mice have a very mild phenotype. So there are dysferlin deficient mice. They've been known for quite some time, and we've created our own. They do develop pathology. So you can see that there's muscle degeneration that starts occurring, after about between 1 to 3 months. You can start seeing, if you look histologically, but they behave, fairly normally out past a year or so, and then you end up seeing some weakness, some kyphosis and things like that. So in terms of looking at like behavioral aspects, it's not until they become much older that they actually start exhibiting some deficits. So that's kind of the downside of these animal models, because it takes some time in order to be able to see that at the behavioral level. But looking histologically, we can see deficits within a few months, 3 to 4 months. You can, and one of the key observations also is, as with many muscular dystrophies, is elevated creatine kinase. So because there's muscle damage, we can use CK assays to readily monitor the earliest aspects of the muscle degeneration. So in terms of modeling and what we would look at, we'll be focusing on the early aspects of histology and creatine kinase assays. But yeah, they do kind of behave, with the patients as well. I mean, it's later onset disease and it's very slowly progressive. And it's just there is just an overall slow degeneration of the muscle and then the accumulation of fat and fibrous tissues that just makes it totally non-functional. Does that answer your question?
It does. I was wondering if you challenge them with like some severe exercise regimen or something like that, if they would show earlier phenotypes. But.
Yeah, people have done that to some extent, and it maybe offsets it a little bit, but it's not like it drives them, like it doesn't make them crash. So, and except at least at the younger ages. So,
Larry.
Yeah, great presentation, Janice. I just have a really quick question. So, sure. The detection of these pseudo exons is really interesting, and I'm just wondering how feasible it may be for patients where whole exome sequencing, doesn't give an answer. Would it be reasonable to do long read sequencing of RNAs targeted to known muscular dystrophy genes and just detect the variants directly?
Yeah, so I firmly believe that's to be the case. Actually, having found these two, quite unexpectedly. Yes, I think looking at the RNA sequencing and splicing patterns is a really good way to, once you've kind of like exhausted the obvious exon patterns. And in fact, we have some collaborators that are going to be sending us some additional patients now. So once this is set up and you can actually know what you're looking for, I think seeing the sequence of the RNA is much more telling. I think to look for these kind of aberrant splice forms. Even if you just look sometimes for at the RNA-seq level, you may kind of miss it. So the long splice, actually looking at the long reads through the actual messenger RNA, I think is a better way to do it. So at least for us. So yeah, and they're probably more out there. I mean, so and this is the whole concept of aberrant splicing and cryptic exon splicing is a field that's been known for a few decades now, and there's hundreds of examples of this in various diseases. And so these things exist, but they're not always picked up right away because the first line of attack is the exome sequencing. So I think there'll be a lot more done with this. And that's why this is an exciting model, because it's kind of like a proof of concept type of thing for dysferlinopathy, and to see if we'll be able to modulate this, and it may help other patients.
Thank you so much, Janice. You are passionate. We need to move on.
Okay.
Let's hear Dr. Hayward now.
Thank you. Thank you very much.
Thank you. Thank you so much. So I will let Dr. Hayward, who is a professor of neurology in our department, and it's a clinical scientist, and he is going to show us his slides right now. Yep.
Hello, I'm Larry Hayward, an investigator in the UMass Wellstone Center for Facioscapulohumeral Muscular Dystrophy. Today, I will provide a brief clinical overview of FSHD, discuss some recent findings using cellular models to study the disease, and summarize some new developments toward therapies for FSHD. I am a site PI for the Fulcrum Therapeutics trial of losmapimod in FSHD and a consultant for Myasea. FSHD is the second most common muscular dystrophy prevalent in adults and exhibits both genetic and epigenetic susceptibilities. Patients typically notice weakness of the shoulder girdle and pectoral muscles, which can progress to involve the upper arm, face, especially around the mouth and eyes, and some leg and abdominal muscles. The pattern of weakness is characteristically asymmetrical and can be highly variable, even among family members. Fortunately, eye movements, swallowing, and cardiac muscles are generally spared in FSHD. Shown above is scapular winging, more prominent on the right, and the image below shows a distinctive pattern of supraclavicular fullness due to an elevated scapula, pectoral and humeral muscle wasting with relative deltoid sparing, abdominal weakness, and sparing of the forearm muscles. Facial weakness usually involves the muscles around the eyes and mouth, but can sometimes be very subtle. This can include syndactyl or eyelashes sign here of the right eye with inability to bury the eyelashes completely when attempting to close the eyes tightly. Subtle difficulty raising the corners of the mouth can be seen, or asymmetry when pouting the lips due to right-sided weakness here of the muscles around the mouth. And in addition, patients can have difficulty puffing out the cheeks. The genetic features of FSHD are very interesting but complex. Near the tip of chromosome 4, an ancient set of 3.3 kb repeating units known as D4Z4 occurs, normally with greater than 10 repeats. Each D4Z4 unit contains a copy of the DUX4 gene that encodes a powerful embryonic transcription factor, which normally is epigenetically silenced after early development. The root cause of FSHD is inappropriate de-repression of DUX4 expressed from the last repeat in the context of a polyadenylation signal, or 4QA haplotype, that stabilizes the message. FSHD1 is caused by genetic contraction of the repeat array to fewer than 10 units on a background of the permissive 4QA haplotype, which triggers production of a DUX4 message in a very small fraction of cells that can then be translated to produce the DUX4 protein that is toxic to muscle cells. FSHD2, which makes up 5% of cases and has virtually the same clinical phenotype, is characterized by a low normal sized repeat array of 8 to 20 units and the presence of additional mutations in chromatin modifier genes, also on a 4QA background, causing similar expression of DUX4. DUX4 can activate many downstream target genes that contribute to dysregulation and muscle cell death. Our Wellstone group here at UMass, led by Charlie Emerson, maintains a large biorepository, including muscle biopsy cells from individuals with FSHD and controls. We have asked FSHD patients and their unaffected first-degree relatives to donate blood and muscle samples. From the muscle, the Emerson lab has purified NCAM-positive myoblasts, which can be used in primary cell cultures or immortalized and used to generate induced pluripotent stem cells. We obtain from our human muscle biopsies for preclinical studies, and Charlie Emerson's group has taken human pluripotent stem cells derived from our biopsies and put them through a optimized protocol for first generating myogenic precursors and then myoblasts that are competent to fuse into myotubes in vitro. One drawback of this primary myogenesis protocol is that the cells don't appear to induce the expression of DUX4 as would be the case from the original primary myoblasts. And so Charlie's improved on this protocol to generate what are called induced secondary myoblasts. And these are essentially derived from what we call reserve cells, which are cells that fail to fuse at this stage here, but that can be revitalized with growth factors, expanded, and then show greatly improved transplantation efficiency. So these are being used in transplantation experiments now and show good promise for providing unlimited number of cells derived from our patients. We have recently examined single-cell gene expression patterns from muscle biopsy cells and these reprogrammed cell classes to better define their similarities and differences. In addition, we wanted to compare the behavior of cells from individuals with FSHD and controls. Here we show cell clustering based on principal component analysis of expressed genes from thousands of cells from 3 individuals with FSHD, 15A, 17A, and 30A, and corresponding family member unaffected controls. We observed quite distinct cell clustering based on cell class and more subtle heterogeneities within each cluster contributed by cells from different individuals. In this experiment, using proliferating single cells, we did not expect to see large differences among individuals due to DUX4, because DUX4 tends to be induced in culture mainly in postmitotic myotubes. Using this same clustering, we can focus on expression of specific genes of interest. Here we show that S1 cells are enriched for the PAX3 upstream myogenic regulator and also specifically express Lin28 RNA that plays a role in self-renewal, consistent with their identity as myogenic precursor cells. In contrast, the proliferating biopsy-derived myoblasts expressed classic muscle regulatory factors such as MyoD and MIF5, some of which were also present in maturing S2 cells, while the ISM cells exhibited a less differentiated state consistent with their increased proliferative capacity. S2 cells exhibited a gradient of developmental progression with muscle differentiation markers such as myogenin and perinatal myosin. The most differentiated of these cells adopted an elongated shape, also characteristic of cultured myoblasts, becoming competent to fuse into myotubes. We also observed relatively specific expression of genes such as RGS4, a regulator of G protein function in ISM cells, or NFIX, a regulator linked to fetal stage myogenesis in primary biopsy cells. Our analysis of thousands of genes further defined heterogeneity across cell classes and also captured subtle differences between the individual cell donors. Comparing transcriptomes using pathway analyses has allowed us to determine differentially expressed gene sets for each pairwise cell class comparison. A surprising finding was the detection of rare DUX4 transcripts and its target genes in S1 cells from FSHD donors versus control individuals. Furthermore, the susceptibility to express target genes was highest in cells from 2 FSHD individuals with infantile onset, indicated in light blue, compared to one with childhood onset in green and one with adult onset in yellow. We interrogated these rare cells expressing DUX4 or a core set of its biomarkers to determine additional possible genes enriched specifically in cells upon DUX4 activation. Some of these genes potentially may play a role downstream of DUX4 activation in FSHD. Next, I'd like to discuss briefly several new approaches for developing effective FSHD therapies. Some of the most exciting advances over the past few years have included strategies targeting DUX4 expression or activity, considered the root cause of FSHD. This is being investigated at multiple levels. First, at the DNA level, efforts have begun using CRISPR-Cas9 editing to correct mutations in FSHD2 genes, such as SMCHD1, that are important for normal chromatin remodeling, so that DUX4 epigenetic silencing can be restored. In addition, it may be possible to introduce mutations in the 4QA polyadenylation signal to destabilize any DUX4 transcripts so that they cannot be translated into the active DUX4 protein. Several small molecules that may block the inappropriate expression of DUX4 in muscle are also being evaluated. One example of this is a p38 MAP kinase inhibitor, losmapimod, which has been shown to be safe and well tolerated. We participated in the Fulcrum Therapeutics trial of losmapimod given orally twice a day versus placebo in 80 FSHD patients. The primary endpoint of the trial was a change in DUX4 activity as inferred from DUX4 target gene expression in sequential needle muscle biopsy samples. These were collected from regions of active disease as indicated by STIR-positive muscle MRI signal. The original design was to obtain muscle biopsies at 4 months after randomization, but this was extended to 9 months for 32 of the subjects due to COVID-19 related restrictions. Secondary and exploratory endpoints evaluated changes in muscle fat composition on MRI, clinical measures of muscle function, and patient-reported outcomes. The primary endpoint was not met in that DUX4 target gene expression from small localized muscle samples showed up to 1,000-fold variation in both the treated and untreated groups, so a significant change over time could not be documented. However, there were significant, though very modest, benefits detected for other measures, including decreased muscle fat infiltration in intermediately affected muscles, slightly improved reachable workspace surface area, and improved muscle strength by dynamometry for some muscles. No severe drug-related adverse events were observed, and Fulcrum plans to meet with the FDA before year-end to discuss next steps. One of the major challenges for FSHD clinical trials is that we do not have robust molecular biomarkers to monitor disease severity and progression, which can be slow compared to the duration of drug trials. There's interest in measuring possible cytokine changes in the blood, such as IL-6, which appears to be increased and potentially could be targeted with existing inhibitors. Use of whole-body muscle MRI was informative in the Fulcrum trial, and complementary measures, including muscle ultrasound and electrical impedance myography, are being explored. Innovative clinical measurements, such as the reachable workspace, measured by a camera and mapped in 3 dimensions, as well as other activity-based monitors that can be assessed by patients longitudinally at home, also offer promise for future trials. These and other measures are being assessed in 2 ongoing natural history trials. Several exciting nucleic acid-based strategies to inhibit DUX4 expression at the RNA level are being explored by several groups, both in academia and industry. These therapeutics recognize DUX4 mRNA and bind it, triggering its cleavage or degradation using cellular silencing mechanisms, or sterically blocking its polyadenylation to destabilize the message. Here are 6 approaches presented this past June at the FSHD International Research Congress. The first 4 are RNA interference strategies, which use small interfering RNAs to bind DUX4 RNA and initiate its degradation by the RNA-induced silencing complex. Caitlin Damon in Charlie Emerson's group here at UMass used DUX4 targeting siRNAs conjugated to docosahexaenoic acid to improve delivery to FSHD myoblasts in vitro and to mice engrafted with human FSHD cells in vivo. Lindsay Wallace and Scott Harper in Ohio, also members of our Wellstone team, reported in vivo studies of an AAV-based gene therapy project to inhibit DUX4 with an engineered microRNA called MI405. A single intramuscular injection of AAV6 vectors protected FSHD mouse muscle out to at least a year, and systemic IV delivery of a high concentration improved muscle function out to 6 months. IM injection of AAV6 vectors also reduced DUX4 and 8 of its target genes in the human on mouse xenograft model of the Emerson lab. In another approach, DNA gapmer antisense oligonucleotides trigger cleavage of target duplexes by RNase H. Linda Bowman at Leiden used DNA gapmers mixed with palmitate injected weekly to reduce DUX4 and target genes and showed histological and treadmill improvement in FSHD mice, but no change in muscle weight or grip strength. Finally, morpholino oligomers can be targeted to cells using cell-penetrating structures or coupling to a transferrin receptor antibody to block polyadenylation of DUX4 RNA. Each of these approaches toward FSHD therapies has advantages and limitations, and it will be exciting to follow progress in the area over the next year. Thank you for listening to this update on FSHD modeling and therapeutics. I'd like to acknowledge members of our Wellstone Center team who are contributing to ongoing studies. These include our UMass Neurology clinicians and colleagues listed here, including Charlie Emerson, Oliver King, and members of Charlie's lab I've been very fortunate to work with, especially Dongsheng Guo and Caitlin Damon. I'm happy to entertain any questions you may have.
Thank you so much, Larry. We have to make a decision because we are in the break right now, but I think your presentation was so exciting to hear. If there is anyone that wants to ask questions right now, and we can take 5 minutes break after that. I have a question, Larry. Does the LOSMAPI mode made it on the FDA approval for any other indication? I know that there were phase 3 study on multiple other indications, depression, COPD, and other diseases.
Right, so that agent has been, one of the reasons Fulcrum chose that agent was because it had been studied in other conditions, including COPD, and seemed to be safe even with people with heart and lung issues. I believe what happened with those trials was it just wasn't efficacious in those particular trials. So I'm not aware of it having any other clinical use right now. Bob has a question.
Yep.
Yeah, thank you. Quick question. Can you please comment on why DUX4, when misexpressed, is so toxic?
Oh, yes. So briefly, DUX4 is turned on during early development during the 4 to 8 cell stage. It's important for zygotic genome activation, and it actually turns on hundreds of targets at that time. And so one of the theories is that if it comes on in a mature muscle, it activates many of these other genes. It's a very powerful transcription factor, and it dysregulates gene expression and leads to all sorts of problems, such that when activated in cells in culture, those cells die within 24 hours. Fortunately, it only comes on if you do a muscle biopsy, it's only detected in about 1 in 500 muscle cell nuclei. So there's some trigger that fortunately is only rarely activated, but once it's activated, that cell is gone. And so we need to understand more about what actually is the trigger and how can we prevent that.
Thank you.
I don't see any other hand up, but it's any other question for Dr. Hayward? Okay, so we have a break of 5 minutes. So we are back. We should be back by 2:06. Thank you, everyone. So, because the time is up, I think will be better if we go ahead and resume the program with Dr. Charles Emerson talk, who is a professor of neurology. And he is not with us right now, but one of his collaborator from the lab, Kathleen Daman, is going to answer the questions at the end. So Jennifer, can you help with the talk?
Good morning, everyone. I'm Charles Emerson, professor of neurology at the UMass Chan Medical School. I'm looking forward to telling you all about our gene editing therapeutics for muscular dystrophies. Muscular dystrophies are a wide diversity of muscle diseases that result in muscle weakness. Today, we're going to focus on the limb-girdle type muscular dystrophy. So they're basically these dystrophies are basically classified into 5 groups based on the main areas of muscle weakness. However, muscular dystrophies are caused by mutations in about 30 genes that encode muscle proteins that either connect muscles to the extracellular matrix or are involved in muscle contraction. So being genetically caused diseases, gene editing is really a prime approach to being able to think about therapeutics and develop therapeutics for these diseases. It's also notable that cardiac and skeletal muscles are also affected together in many of the genetic muscular dystrophies because the same genes that are expressed in skeletal muscle are also expressed in the heart. So one day, about 4 years ago, a young man, about 35, came to my laboratory, and he told me he had a muscular dystrophy that nobody else was working on, and he was affected by it as well as his sister. In fact, he was quite disabled. And the dystrophy, he said, was called limb-girdle R7. Now, the truth be told, I'd never heard of this muscular dystrophy. And when I looked it up and talked to him about it, it was a rare autosomal recessive disorder that it affects muscle weakness in the pelvic and shoulder girdles as typical of limb-girdle muscular dystrophies. Onset was during adolescence and progressed to severe disabilities. And he was in his 30s and clearly about to be wheelchair bound. His sister had already progressed further. The cause of death for this dystrophy usually is from cardiac failure because, as I said earlier, many muscular dystrophies affect both cardiac muscle function as well as skeletal muscle function. So I asked him what this limb-girdle R7 mutation was, and he told me. He was very well informed and about his own disease. And he said it's caused by mutations in the TCAP gene, which encodes the protein telethonin. And again, truth be told, I just barely knew a little bit about telethonin protein. Telethonin is a protein that is associated with the muscle contractile apparatus, and as we've shown here, the sarcomere, and it's associated with the Z disc, which is this dense area here on the electron micrograph. And what it also is associated with is a very large protein called titin, which is shown here in red, strings through the thick filaments of the muscle. And this titin is the spring that when muscle contracts, it allows this muscle to spring back again. And so it's a spring protein. And its telethonin's function, which is the R7 mutation, is to couple telethonin to the Z disc. So presumably during these dystrophies, the R7 dystrophy, the telethonin function is compromised, and it compromises the spring action of telethonin, allowing muscles in the heart and skeletal muscle to wear out. Again, this is not a well worked on muscular dystrophy, and of course, that's really important because this is an opportunity for us as clinical researchers to be able to make some discoveries that may have impact on the whole field of muscular dystrophy. So I said, sure, we'll work on your disease. And it was really important. Nobody else is working on it. And, but, you know, and we think we can make some progress because we developed a platform for investigating this disease called an IPS therapeutic development platform. And what this will require you to do, speaking to the patient, is to donate some skin cells to put in cell culture for us that we can reprogram with reprogramming genes that will make your adult cells into embryonic iPS cells, induced pluripotent stem cells, that then we can induce to form muscle and muscle stem cells by a technology that my lab has been pioneering. And using these muscle stem cells from your disease, your body will be able to study and develop disease models for the muscular dystrophy you have, and those models can be used for therapeutic development. But also, we're able to use your cells converted to muscle stem cells for gene editing and stem cell therapeutics, and which is what I'm going to speak about today. So he agreed to give his skin cells, which we cultured, and then we reprogrammed them into iPS cells and made them into muscle cell lines, myoblast stem cell lines for doing gene editing and stem cell therapeutics. And so we did this reprogramming and derivation of muscle stem cells from this patient in about 4 or 5 months and accomplished the goal. In the meanwhile, we started to put our heads together to figure out, well, how are we going to treat this disease? How are we going to repair the mutation in the telethonin TCAP gene? So first of all, we want to understand what the nature of the mutation was. And it's a founder mutation. He was from Australia originally, and but ancestrally from northern China, where this disease is much more prevalent. It turns out that the mutation is a microduplication in the telethonin TCAP gene in the coding region shown here. So here you can see CGA, GGTGT is repeated in, so it's a mutation that causes a repeat. And what that repeat mutation does is introduce a stop codon near the N-terminus of the telethonin protein that allows the protein not to be expressed in muscle. So, and that's why it's an autosomal recessive disease. So we understood the mutation was a microduplication. So I got together with my colleague, Scott Wolf at UMass, who have been working on gene editing therapeutics for other muscular dystrophies. And we thought about how we might attack correcting this mutation. And he remembered some minor literature about a repair pathway, a DNA repair pathway that had been described in people called microhomology-mediated end joining. And this repair process allows, when you have microduplications, when you have DNA double-stranded breaks in these duplications, this mechanism of microhomology-mediated end joining, MMEJ, allows for the filling in of this microduplication and correction of the microduplication, making it into removing the microduplication. So that is sort of was in the literature. So we reasoned that if we use the CRISPR enzyme, Cas9, which makes double-strand breaks in the DNA, coupled with a guide RNA, which will bring this enzyme to the microduplication in the middle of the microduplication. The guide RNA is associated with the Cas9 enzyme, and together the guide RNA brings the enzyme to this microduplication and create a cut in the microduplication. Then microhomology-mediated end joining then might correct the mutation So that was our hypothesis. Totally speculative, but based on this limited knowledge of MMEJ and the use of Cas9 guide RNAs to target very specific DNA sequences in the human genome. So we had made the cells that from the patient, and made myoblasts, these stem cells from his iPS cells, is shown here. This is a nuclear stain. And then, so what we did as an experiment, we added a Cas9 protein plus the TCAP guide RNA that would guide to the microduplication, and packaged them up into RNP particles, that's ribonucleoprotein particles, added them to our tissue culture cells, let the cells differentiate into muscle. You can see these nice muscle fibers in culture. And then we asked whether this approach really would resolve the mutation by MMEJ. So that required doing sequencing of the TCAP gene to see if the mutation had been corrected, and also looking to see whether telethonin protein had been restored. So we were just amazed at how well this worked. Normally, CRISPR technologies for gene correction are very low frequency events, very low frequency, because they require homologous recombination and very, very fancy maneuvering of DNA to get the correction to happen. So what we observed when we sequenced this allele, that 68% of the cells had a perfect correction of the locus. In other words, he had now resolved back to what a normal locus would look like, no microduplication. And so a major proportion in iPS cells and in myoblasts derived from the patient, we got this very effective correction, perfect correction. And also there was some imperfect corrections created by cutting, they're called indels, when you cut this repair machinery that then does a little bit of repairing of the cut site, leading to in-frame indels. These would be coding protein, but not exactly the identical protein to telethonin, but with small amino acid changes in the region of the mutation. So this was amazing. So we actually had made a discovery, a really interesting discovery. And it turns out that if you look at protein expression for telethonin, yes, when you do the gene correction in these patient-derived stem cells and they become differentiated muscle, yes, you increase the expression of telethonin protein. In other words, you restored the protein, which is the whole purpose of this work. So this was very exciting for us. We made a discovery from this patient, working with this patient on a new mechanism for gene editing called microduplication editing with MMEJ. And it was even more interesting because our colleague, a bioinformatics colleague, Oliver King, then looked in the genome to find that there are at least 143 disease-causing microduplication mutations in the literature, in the clinical database for disease-causing mutations. And they're either neurological or muscle. And in fact, some of these mutations are rather substantial in the population, such as the mutation causing Tay-Sachs disease. So this seemed to be a very broad discovery that had implications beyond the R7 muscular dystrophy. And this led us to publish a nice paper in Nature. So what do we need to do next towards the therapy? Well, we need to move the therapy in vivo to treat patients, and we needed to do this in an animal model. So we developed an animal model for delivery of Cas9 and guide RNA to the TCAP locus. We created a humanized TCAP mouse. So this mouse, what we did, we converted this mouse TCAP gene into a human gene with a human mutation for R7. So we did that by synthesizing about a 40 base pair piece of DNA around the mutation that included the mutation, used CRISPR editing in embryos, early embryos of the mouse, by homologous recombination replacement. We put the human sequence in place of the mouse sequence for TCAP, and then we made germline mice. They do have the muscular dystrophy that the patient had. They get muscle weakness with aging and muscle turnover with aging. So we had made a mouse model that we could then do correction with. So, and that work was done by Greg Cox at Jackson Lab, a collaborator of ours who made the mouse. So look at this nice mouse that Greg had made. So here's a wild type mouse, and here using an antibody to telethonin, you can see how this nice cross-striated immunostaining of works for telethonin in a wild type normal non-diseased mouse. Nicely organized protein expression in the sarcomere as we described where telethonin should be. In his TCAP mutant mouse, in the muscle fibers, there was no immunostaining with the telethonin antibody. There is some background staining in the matrix, which often happens with muscle antibodies because it's such a dense tissue, but there's no staining of telethonin in the muscle fibers. So again, he had successfully made a knockout mouse, a mouse with a human mutation that actually created a deficit in telethonin expression in vivo in muscle fibers. So then we attempted to rescue this mutation in vivo using an AAV delivery of Cas9 and the TCAP guide RNA. So there are AAV vectors which will infect muscle tissues throughout the body. And we used a very specialized one developed by Stephen Hauschka at Seattle, and Eric Olson used this at UT Southwestern and gave us the vector. And what is so unique about this vector is that it drives the expression of Cas9, the protein that's going to do the double strand cut in the microduplication, under the control of a muscle-specific promoter. So it's only expressed in heart and skeletal muscle. So perfect that we can actually express the Cas9 just where we want it when we infect the muscle cells and the cardiac muscle with this AAV virus. And then we also used a TCAP guide RNA, a second vector AAV with a different promoter, more generalized promoter that expresses the guide RNA for TCAP, directing to the microduplication, also expressing a turbo RFP, that's a color marker that'll tell us whether we've got infection of the muscle. So we did this experiment and got a fantastic result. Excuse me, where we had where about 60% of the muscle was actually now TCAP positive, telethonin immunostain positive when we infected these mice. And you can see this is the red of the guide RNA. And this is the green of the telethonin staining. Not all fibers are stained, but 60%. And you can see the cross striations in there that it actually restored telethonin expression in about 50 to 60% of the muscles we used 2 AAV vectors. And here's a large image of one of these muscles, which you can see the extent of the gene of the therapy that we've got here for telethonin restoration of function. This is extremely encouraging. However, now comes a surprise. So when we did the DNA sequencing of these mouse muscles, we found very surprisingly that there was no perfect correction, very little perfect correction of the microduplication. But there were many indel in-frame mutations that occurred. These are small mutations that changed 1 or 2 amino acids in the region of the original microduplication. And that is what we think is resulting in the expression of telethonin in these muscles. So our idea about microduplication collapse and resolution through MMEJ does not work in adult muscle. But we do get telethonin restoration very efficiently through a second mechanism of indel in-frame mutations. So that was, a very unexpected, but very potentially encouraging result. So what have we learned? So we're in our way towards the clinic for this patient that came to see us. Well, when we began to move our discovery of MMEJ gene therapy, gene correction in vivo, we found that the mechanism for MMEJ did not work in differentiated muscles, but we were able to get effective correction of the TK mutation through this indel mechanism. So we're now doing experiments to see whether the small amino acid changes in the telethonin proteins caused by indel mutations is enough to restore muscle function in the TCAP mouse. And our structural work suggests that this correction mechanism will work to give functional protein. So that's very encouraging towards the clinic. We also discovered in our work in tissue culture that we can correct muscle stem cells in vitro in tissue culture by the MMEJ mechanism. So we're now doing experiments in vivo to see if we can use the gene editing approach we have to correct the microduplication by MMEJ in muscle stem cells, which would be a very exciting clinical approach because having the stem cell population corrected will allow long-term restoration of muscle with corrected stem cells for a long-term therapeutic outcome. We're also developing alternative gene editing strategies to correct the TCAP microduplication precisely. For instance, prime editing, which is a newly developed mechanism for gene editing, would be perfect for this. And so the toolbox for gene editing is very deep now and allows us to really attack mutations, all kinds of mutations in very specific ways. So as an alternative to MMEJ, we're developing the prime editing approach to correct the TCAP mutation perfectly. And finally, we're developing technologies for the delivery of edited myoblasts into muscle in animals and hopefully in people doing stem cell therapeutics. In other words, we know we can correct the TCAP mutation in muscle stem cells. We know these stem cells are engraftable in mice. And so the long-term goal would be to use our editing, MMEJ editing for stem cells in combination with a stem cell transplantation approach to do correction in patients through a stem cell therapeutic. So this work started with this one patient and we're still hopeful we'll be able to develop therapeutics for this patient by gene editing. We've also provided our mouse, our gene-corrected mouse with a human, our mouse with the microduplication mutation to other researchers who have gene therapy approaches, other approaches for treatment of muscular dystrophies. So we're hopeful that this patient will, in the next few years, be part of a clinical trial to correct his mutation. So this is an N of 1 right now, but we are very hopeful that this therapeutic approach that we're developing for gene editing will impact his disease. Remember that there are over 30 other diseases where mutations occur in muscle proteins that could be treated by muscular dystrophies. So curing one of these diseases with gene editing will really open the door for treatment of many of these diseases using the same kinds of technologies that we're developing with the R7 mutation. So again, the people that did this work, Sukanya is a postdoc in Scott's lab, and Dongsheng and Caitlin are postdocs in my laboratory, did the bench work for this. Oliver did the bioinformatics, showing that many, many diseases are caused by microduplications that could be corrected by the MMEJ mechanism. And Larry is our clinical researcher and clinical scientist who runs the MDA clinic at UMass and can tell you and made the cells from our patient for IPS generation and has been very much involved in this work. So thank you very much. Caitlin Damon will be available to answer questions.
We thank so much, Dr. Emerson, for his wonderful presentation with this, innovative way to do neurotherapeutics and pioneering so much, unique, I will say, gene editing techniques. For the sake of time, I will ask if anyone has a question for Kathleen, to be short question, and Kathleen can go ahead and answer. Thank you for being with us, Dr. Deming.
Sure. Glad to be here.
I have a quick question. I was just unclear about how the indels were affecting the gene and the protein. Have you guys mapped that out? Is it affecting the duplication?
Yeah, so the indels are still at the duplication site. So it's either like a +1 or a +2 nucleotides. And so based on some small playing around with the structure of the protein, when you add in those two sites, it doesn't seem to be disrupting the structure too much. Again, we're just doing this in silico, so we're not actually testing what it is, but it looks to be enough to maintain or to restore TCAP TCAP expression. And we're trying to figure out a functional assay here where we can see if restoring TCAP expression in, let's say, 60% of the muscle is sufficient to do restorative muscle function. So Charlie didn't mention, but all of these experiments we did were intramuscular injections into the TA muscle. So currently we're just doing TA injections. And so it's kind of hard to see TA restoration. So we're hoping to do a more systemic treatment to get all of the muscles in the body, and then we can do a more muscle function restoration test. But we haven't gotten to that point yet. But at least in silico, it doesn't look like these the couple of mutations that are most prevalent after the correction have any sort of effects on the structure.
Thank you. It's very exciting work.
Thanks.
Any other question? Thank you so much again, Kathleen, for being with us and for this wonderful work.
Glad to be here. Thank you.
So now I have the pleasure to introduce one of my collaborators for this program that helped me a lot with bringing you guys together from the labs and clinical. And I hope everyone has a good time. I am extremely excited with all this superb, innovation in treatments that probably are available only at UMass right now. And without further ado, I want to allow Dr. Bosco, who is a professor of neurology, to go ahead with her talk.
Good afternoon. My name is Daryl Bosco. I'm in the neurology department at UMass Medical School, and I have no disclosures for my presentation today on elucidating mechanisms underlying neurodegeneration using human-induced pluripotent stem cells. So most of our work is in the context of amyotrophic lateral sclerosis, which is a neurodegenerative disease that primarily targets motor neurons, causing these cells to degenerate and for patients to become paralyzed. Patients generally succumb to ALS within 3 to 5 years of their diagnosis. And although ALS is not as prevalent as other neurodegenerative diseases such as Alzheimer's disease, there's still about 30,000 individuals living in the United States with ALS at any given time. And the lifetime risk is also pretty significant at 1 to 500 to 1 to 1,000. It is noteworthy that there are overlapping clinical and pathological features with another disease, frontotemporal dementia. So these are the genes that have been implicated in ALS, and they encode proteins that are involved in protein homeostasis that are in green, also RNA binding proteins in purple, and cytoskeletal proteins that are in orange. And so if you overlay these proteins into this illustration of the different pathways and processes that are highly implicated in ALS, the picture becomes somewhat overwhelming. And so I won't go through all these different pathways and processes, just to give you a sense that there are a number of factors that are involved in ALS. It's a very complex disease. And although motor neurons are primarily targeted in ALS, other cell types within the CNS environment also play a role, notably these different types of glial cells. And so in my lab, we've been looking at these several ALS and FTD-linked proteins that are highlighted here. And today I'd like to tell you about our results on FUS, fused in sarcoma, and profilin 1. And so I'll start with FUS, which is a multifunctional, multi-domain protein, which is illustrated here. It plays many different diverse roles in RNA and DNA processing. So any function you can think of that involves RNA or DNA processing, FUS has been shown to have a role in that pathway or process. And so it's been difficult and challenging to understand how ALS-linked mutations affect FUS function and cause ALS. And so we've been trying to tackle this question using human-induced pluripotent stem cells, which we can differentiate into different cell types, including spinal motor neurons. And so the premise here is that different cell types like fibroblasts or other somatic cell types like blood can be reprogrammed into a pluripotent stem cell state. And then using different small molecules or by forced expression of different transcription factors, we can force these cells into different cell types, again, like neurons or many other different cell types, all depending on the type of molecules or transcription factors that are added to the cells. And so some advantages of the IPS system is, first, we can look at human cells, so they're more relevant to human disease. And also the genes are expressed at endogenous levels. And so there's less manipulation in terms of overexpression of proteins in these cells. So they represent a relevant and strong model system for studying neurodegeneration in the lab. So this is a diagram of what we've done in my lab. Is we've used this small molecule approach where at the indicated days over a 2-week period, we add these drugs, these different small molecules, and this forces the iPS cells into ultimately a motor neuron fate or state. And so looking at FUS, what we found is that in expression in the in control iPS cells, we see that FUS is predominantly expressed in the nucleus, which we expect, but the ALS-linked mutations, most of which occur in the nuclear localization sequence of FUS, cause FUS to mislocalize from the nucleus into the cytoplasm. And you can see that here, this is a mutation that very strongly abrogates that nuclear localization sequence. So it prevents FUS from being localized into the nucleus. And if we look out in the neurites away from the cell soma, you can see there's a lot more FUS outside in these neurites and these processes compared in the mutant cells compared to the control. And I just want to point out that this the disease severity correlates with the extent to which mutant FUS is mislocalized into the cytoplasm. And so this is an iPS cell line, and these were derived from an individual who was diagnosed with ALS as a teenager. And so this, it's clear that FUS has this impaired nucleocytoplasmic balance. And so we were wondering, and actually a former graduate student in my lab, Nora, wondered whether mutant FUS also affected the nucleocytoplasmic transport pathway. And so nucleocytoplasmic transport, as the name implies, is the transport of biological molecules between the nucleus and the cytoplasm. And this largely occurs through these nuclear pore complexes, which are illustrated as these baskets that are embedded in the nuclear envelope. And these nuclear pore complex baskets contain multiple copies of nuclear porins, these small proteins that are also referred to as NUPs. And so we know that the process of nucleocytoplasmic transport declines naturally as a consequence of physiological aging, but we've also come to realize that in the context of neurodegenerative disease, most prominently in the context of ALS and FTD, this pathway seems to be playing an important role in disease pathogenesis. Also, this has been seen in other neurodegenerative diseases and other neurodevelopmental disorders. And so Nora started looking at this pathway in her iPS-derived motor neurons, and one of the first things she did was to stain or probe for nuclear porin proteins using immunofluorescence. And so these are the nuclei of the iPS-derived neurons, and here we're looking at POM121, which is a nucleoporin that's a scaffold nucleoporin or a scaffold NUP. And what you can see is that the signals for this protein are diminished in the mutant FUS nuclei compared to the control. And we looked at some other nuclear porins or NUPs, including NUP62, which is a central channel NUP, shown here in green. And we can also see that these signals are diminished here. And so she quantified the diminishment of the signal in terms of the number of breaks you see in this circular signal. And you can see that there's more breaks or interruptions in the nuclear pore signal in the mutant cells compared to the control. And so we also looked at active nucleocytoplasmic transport using a shuttling reporter. This is TD tomato, which is a fluorescent molecule that's fused to a nuclear export sequence as well as a nuclear localization sequence. And so the idea is that this reporter is shuttling back and forth between the nucleus and the cytoplasm in our neurons. And we can photobleach the signal in the nucleus and watch that fluorescence recover. And the fluorescence in the nucleus recovers from these fluorescent molecules out in the cytoplasm being imported into the nucleus. And so basically these fluorescence recovery curves reflect nuclear import. And what we see is that the rate of nuclear import is decreased in the mutant FUS neurons compared to the control neurons. And so, these data, and we have other data that have been published in this paper, really show that expression of mutant FUS is sufficient to impair nucleocytoplasmic transport. The active transport and as well as a phenotype that involves diminishment of nuclear pore proteins around the nuclear pore complexes. And we have been working on elucidating the mechanism for how mutant FUS has this effect on this pathway. And what we found is that the mutant forms of FUS engage in these aberrant interactions with the nuclear pore proteins out in the cytoplasm. And we hypothesize that these aberrant interactions prevent NUPs from properly incorporating into the nuclear pore complexes, and that's what causes this decline in transport. I also want to point out that these aberrant interactions between mutant FUS and the nuclear porins were substantiated in Drosophila models of ALS FUS with our collaborator Uday Pandey, who's at the University of Pittsburgh. And this pathway now is a therapeutic target for ALS as well as other diseases too. And there's the development of the selective inhibitors of nuclear export, or these SINE compounds that are being pursued for treatment of ALS. And so in the last few minutes, I wanted to talk about another protein that we're working on in the lab, and this is profilin-1. Mutations in profilin-1 were linked to ALS and were identified by my next-door neighbors and colleagues, John Landers and Bob Brown. And so profilin is best known for its role as an actin-binding protein. Together with these other cytoskeletal proteins, they form a ternary complex that function to synergize and promote efficient actin elongation, which is shown in this cartoon. And my lab has been working on understanding the effects of ALS-linked mutations on profilin structure and conformation, which we published here. And the take-home message of these papers is that these mutations cause profilin protein to become destabilized and misfolded. So they adapt an aberrant conformation that also then leads to defects in actin polymerization. So we did a lot of this work in vitro and using immortalized cell lines. But we, again, wanted to know, well, what are the effects of mutant profilin in ALS-relevant cell types? And so, again, we can take the iPS cells harboring mutations in profilin, and we did differentiate these into motor neurons, but we didn't see any obvious effects on neuronal health, axon outgrowth, neurite outgrowth, things that you would associate with a cytoskeletal protein like profilin. So then we came to recognize that actually profilin is more highly expressed in microglia compared to neurons, and that's true for other ALS and FTD associated proteins or genes. And microglia are proteins that are phagocytic, and actin dynamics is very important for microglia function. So here, this is a video of microglia engulfing some material, and you can see that as they're undergoing phagocytosis and engulfing material, they're moving around, they're very motile, and that movement requires effective actin reorganization and actin dynamics. So just to remind everyone, microglia are resident immune cells of the central nervous system, and they're also important contributors to neurodegenerative disease. Normally, they play homeostatic functions involved in synaptic pruning, phagocytosis, and clearing of debris. And they also respond to injury. Although all these functions become impaired or pathogenic in the context of neurodegeneration, where these cells can become pro-inflammatory and hyperactivated, causing them to secrete toxic cytokines and reactive oxygen species. And so using a different type of protocol, we can take the iPS cells and differentiate these into microglia-like cells. And so again, we can start at the same stage that I showed you before for FUS, but here we use different small molecules to direct them into microglia. And our induced microglia-like cells look and function like microglia, so they express microglia-specific and myeloid-specific markers, which we show here with immunofluorescent staining of the cells. And they also secrete cytokines in response to lipopolysaccharides. So they are reactive to LPS, and they secrete these cytokines over time, as expected. So we've also tried to measure the phagocytic activity of microglia, and we do this using an assay where we take synaptosomes and we label them with pH roto, which is a pH-sensitive dye. And so this dye only becomes fluorescent when it's in a context of a low pH environment. And so just to show you what that would mean in the context of microglia, what we have is here these pH roto labeled synaptosomes would represent the food that we feed to the microglia. They bind through these receptors and they get engulfed inside the cell, and they go through this pathway. They enter into these phagosomes, which then fuse with lysosomes. And the lysosomes have a low pH, and that's because the low pH activates enzymes that degrade the debris that's in these phagolysosomes here. So we did this assay using the pH roto dye, and you can see pH roto labeled synaptosomes as a material or the food that we give to the microglia. And you can see that they do indeed turn red over time, which is telling us that they're engulfing and trying to digest the synaptosomes through that pathway I showed on the previous slide. But what we noticed was consistently that the mutant microglia in red consistently show a higher level of red signal over time compared to the control. And so we don't know if this means that the microglia, the mutant microglia, are just engulfing, they're taking up more of the synaptosomes, which causes them to have a consistently higher amount of red signal over time, or if they're digesting less, that they uptake the material, but then they don't clear it through that pathway. And we're also wondering whether this represents a profilin loss of function or gain of tox function. So I won't have time to go into the details today, but what our data show is that there is impaired digestion in these cells. And so there's something in this pathway that's either become clogged, it's certainly dysfunctional. And so what we see is when we give the synaptosomes to the mutant microglia, they're unable to push it through this pathway and lead to the degradation, which can also cause the microglia to become reactive. And this has important implications for ALS and other neurodegenerative diseases where these cells are really important for clearing dead cells and debris. We are using a small molecule approach to see if we can target this pathway and rescue this phenotype. In terms of the mechanism, we think that this could represent a loss of function mechanism, because when we look at profilin levels in these iPS microglia, we can see that the levels of profilin are much lower, significantly lower in the mutant cells compared to the control. So in summary, in our lab, we are using iPS cells because they appear to be a good model for investigating mechanisms of mutant genes using disease-relevant cell types and recognizing that there are multiple cell types that contribute to neurodegenerative diseases. Going forward, we aim to elucidate how FUS and NUP interactions lead to impaired nucleocytoplasmic transport. Within the field, people are targeting the mutant FUS gene, including here at UMass. And also there are the SINE molecules that are being used to target the nucleocytoplasmic transport pathway. we're also aiming to understand how ALS profilin impairs this endolysosomal pathway. And in my lab, in collaboration with others, we're trying to identify small molecules that can stabilize the mutant protein. I wouldn't recommend knocking it down since it's already has decreased expression under steady state. So probably don't want to use ASOs or microRNAs against profilin. But we could use small molecules that stabilize a protein, and we also can use small molecules that can enhance autophagy and clearance of the debris through that path. And finally, I would just like to thank my lab. This is a current picture of my lab and my lab members, my collaborators outside of UMass and within UMass, and then also my current funding sources. And thank you for your attention.
Thank you so much, Daryl, for this very elegant, work, easy to follow, well done. At this moment, I think it's important to have a few questions. I don't think we have time for the break, but the good news is we just had a break. So I think we can, get more questions and answer from Daryl until 2:55. So we have 4 minutes. Daryl, I will ask you, have you thought about trying to see if you can have some IMGs, microglia derivates from other diseases that don't have anything to do with the genetic, and see if you, so the experiment, it's completely opposite what you have done, to see if there is any kind of suggestion of having the profilin functional, or dysfunctional present in autoimmune diseases, let's say, or Alzheimer or something else.
Yeah, I hadn't thought about looking at profilin so much in other models, although it could be. I mean, there's some work coming out now, although mutations in profilin are pretty rare in ALS. It does look like there's pathogenic profilin species and sporadic disease and sporadic ALS. So it could be that profilin is dysfunctional in more than just and familial cases of ALS. And looking at non-familial cases of profilin, like generally in the IPS system, it's hard to look at sporadic cases. And I think that's true for most neurodegenerative diseases because already there's so much heterogeneity. And the IPS system it's difficult in terms of reproducibility. So the controls have to be very, very tight. So we have mutant control, we have mutant cells, and then we have isogenic controls, which in theory should be exactly the same, except that they have a mutation. Once you have sporadic, there's no defined mutations that you're looking at. You just have, heterogeneity, genetic heterogeneity. And so I think it's very hard to see phenotypes when you have that kind of situation, because if it's variable, you don't know if it's a real phenotype or it's just the cells aren't behaving properly and just the data are a little bit all over the place. So, so we've really been focused for all the IPS work on familial mutations in ALS. And I do think that profilin plays a normal and important role, and other people have shown this in microglia and phagocytosis autophagy. And so it could be that it's playing a role in these other diseases. I actually hadn't really considered looking at profilin specifically in other disease microglia, but that's an interesting thought because I do think it plays a normal function, and maybe it is messed up there too in other diseases. Thank you for that question.
Yes, Brian.
I guess along the same line, given the work that Dori Schaefer is doing with microglia, I was wondering if there's some overlap in the works that you're both doing, given the fact that you found this new avenue for microglial function or dysfunction.
Yeah, I have to credit my work on my interest in microglia to Dori, because actually when soon after she got to UMass, she started talking to us about microglia, and these protocols that came out for differentiating iPS cells into microglia are relatively new. So this project started as a Riccio funded grant, a Riccio fund grant. that Dory and I both had together. And so she was the microglia expert. I was like the IPS person. And so we sort of put those two things together, and we developed these cells. And so I think that in the microglia field, there's a lot of skepticism about looking at cells in vitro. So that microglia, once you take them out of an in vivo context, they change a lot. So there's a lot of concern about focusing on microglia in vitro, which we're finding now that we're submitting grants to NIH and things like that. But I think our approach is to still use the IPS system, because if it's in a cell and in a dish, you can really do a lot in terms of manipulating in your experiments that you can't necessarily do in mice. But we are complementing these studies with mice, and we have CRISPR mice from John Landers that express the same mutations in profilin that we have in our iPS cells. So we're trying to use both in vivo and in vitro models together.
Excellent. Thank you so much, Daryl.
Thank you.
And now it's time to hear our interim chair, Dr. Brian Silver, who is a professor and, how I said, interim chair for our department. And he has helped us as a co-director of this program. And I want to thank him for the opportunity to have this marketing by hiring Jennifer. It's wonderful to have this ability to, have more visibility for our department. Brian, go ahead.
This is a presentation on tenecteplase versus alteplase for acute ischemic stroke. These are my disclosures. So the objectives for this talk are to enumerate potential advantages of tenecteplase in acute ischemic stroke, contrast the differences in dosing and administration between alteplase and tenecteplase in acute ischemic stroke, and identify the current evidence to support alteplase and tenecteplase in acute ischemic stroke. This is the mechanism by which activase and alteplase produce a fibrinolytic effect. TPA converts plasminogen to plasmin, and therefore leads to degradation of fibrin into fibrin degradation products. The differences between alteplase and tenecteplase are summarized in the slide below. Alteplase is a direct plasminogen activator, as is tenecteplase. Tenecteplase is slightly more fibrin specific than is alteplase. In terms of half-life, alteplase is 5 minutes and tenecteplase is 20 minutes. Dosing is 0.9 milligrams per kilogram with alteplase versus 0.25 milligrams per kilogram with tenecteplase, except for Norway, where the dosing is 0.4 milligrams per kilogram in studies. Tenecteplase is administered as a single bolus over 10 seconds with a maximum of 25 milligrams versus alteplase, which is administered as a bolus of 10% with the remaining 90% as a bolus over 60 minutes and a maximum of 90 milligrams. The PAI-1 resistance is low with alteplase and 80-fold higher with tenecteplase. And in terms of genetic alteration relative to native TPA, alteplase is a recombinant form of TPA, whereas tenecteplase is a genetically modified form of TPA. In terms of the current indications for the various lytic agents, alteplase is approved for use in acute ischemic stroke, pulmonary embolism, and STEMI, whereas tenecteplase only has an approved indication in STEMI, and this may be a barrier in some institutions where a conversion from alteplase to tenecteplase can be an issue, particularly for organizations that want to preserve alteplase as a treatment for acute PE. So the potential advantages of tenecteplase over alteplase are better clot lysis due to greater fibrin specificity, Potentially faster door in, door out times for satellite hospitals due to not having to wait for ambulance with personnel who can manage a drip. Faster door to TPA times due to not having to prepare an infusion, and potentially less costs compared to Alteplase, which currently is about $7,800 versus $6,000 for Tenecteplase. If using 340B pricing, then the difference is much less, perhaps $100. Potential disadvantages of Tenecteplase: there is a need to stock 2 lytics in the emergency department for different conditions, and in particular, Alteplase is the only recognized drug for pulmonary embolism. Second, dosing errors may occur during the conversion period, that is, a 0.9 milligram per kilogram dose of Tenecteplase may inadvertently be administered instead of the required 0.25 milligram per kilogram. And currently, Tenecteplase is not reimbursed if wasted by the manufacturing company compared to Alteplase, which is reimbursed. So these are the currently completed trials, and I will go through some of these in the subsequent slides. As you can see, most of the studies used 0.25 milligrams per kilogram of Alteplase, with the exception of the Norwegian trial called NORTEST, and in the Extend IAT-NK Part 2 study, they compared the 2 doses of Tenecteplase. Most of the studies were done in less than 4.5 hours from onset, and in general, the studies either showed an advantage of Tenecteplase over Alteplase or no difference between the 2 drugs. So we'll start with this first study, which was performed by Parsons et al. In Australia. And this figure shows that in terms of reperfusion rates, there was a greater reperfusion effect with Tenecteplase at 0.1 milligram per kilogram, and even greater with 0.25 milligrams per kilogram as compared to Alteplase at the standard dose of 0.9 milligram per kilogram. And in terms of NIH stroke scale scores, there was an advantage of Tenecteplase at 0.1 milligram per kilogram and 0.25 milligram per kilogram with respect to reduction in NIH stroke scale scores over time, which implies the patient is improving as compared to Alteplase at 0.9 milligrams per kilogram. In this study, Tempo 1, performed by Coutts et al. In Calgary, Canada, they compared a dose of 0.1 milligram per kilogram versus 0.25 milligrams per kilogram in patients with low NIH stroke scale scores and a large vessel occlusion who did not go for thrombectomy. In this case, they found that 0.25 milligrams per kilogram had a favorable advantage over 0.1 milligram per kilogram with respect to modified Rankin scale score outcomes at 90 days. And there is an ongoing study comparing 0.25 milligrams per kilogram versus placebo in these patients. This study, called ATTEST, was performed in Asia and surrounding continents. In this study, in contrast to the study by Parsons and others in Australia, they did not find an advantage of Tenecteplase over Alteplase in large vessel occlusion. However, they did not show that Alteplase was superior either. In this study called NORTEST, which was performed in Norway, this was comparing a dose of 0.4 milligrams per kilogram of Tenecteplase versus a dose of 0.9 milligram per kilogram of Alteplase, and similarly did not show a benefit of Tenecteplase over Alteplase either in the intention to treat analysis or the per protocol analysis in 90-day modified Rankin scale scores. This study, called Extend IAT-NK, was performed by Campbell and colleagues in Australia and compared using Tenecteplase versus alteplase in patients undergoing thrombectomy within the 6-hour window. And in the study, they did show that tenecteplase produced better outcomes in advance of thrombectomy as compared to alteplase when looking at modified Rankin scale scores at 90 days. In a Part 2 study, they then compared whether there was an advantage of 0.4 milligrams per kilogram versus 0.25 milligrams per kilogram in these patients, and they did not find any advantage of the higher dose, and reasoned that 0.25 milligrams per kilogram in patients within 6 hours undergoing thrombectomy was all that was needed. These are currently the pending trials. The TASTE-A trial is comparing tenecteplase at 0.25 milligrams per kilogram versus alteplase at a dose of 0.9 milligrams per kilogram administered within the ambulance using a CT on board the ambulance, and results are expected either at the end of this year or next year. The TWIST trial has recently completed, just short of 600 patients, and this is a study administering tenecteplase 0.25 milligrams per kilogram versus usual care in patients waking up from stroke but within 4.5 hours of awakening, and results are expected to be presented at the International Stroke Conference in 2022. The timeless study is almost near completion as well. This is a study looking at patients with large vessel occlusion in the 4.5 to 24-hour window who receive either tenecteplase 0.25 milligrams per kilogram or placebo, and comparing outcomes at 3 months, and results are similarly expected next year. The other ongoing studies alluded to previously include TEMPO-2, which is looking at administering tenecteplase 0.25 milligrams per kilogram versus alteplase 0.9 milligram per kilogram in people with large vessel occlusion and low NIH stroke scale scores in a period of 12 hours or less. NORTEST-2 is the Norwegian trial, which is similarly going to look at tenecteplase at a dose of 0.4 milligrams per kilogram versus alteplase 0.9 milligram per kilogram. But in contrast to the original NORTEST trial, which had stroke scale scores in general less than 5, they will be targeting patients with a score of greater than 5 because in their studies, subgroup analyses, they found that potentially that might be a group which benefited to a greater extent. And finally, ATTEST-2 is looking at patients with non-large vessel occlusion, stroke less than 4.5 hours, and administering 0.25 milligram per kilogram versus alteplase 0.9 milligram per kilogram, and results are expected in 2025. In terms of real-world experience, this was the first large-scale world experience just published this month in the journal Stroke, and it compared the experience in New Zealand across a regional stroke network when switching from alteplase to tenecteplase. There were a total of 555 patients before the switch treated with alteplase and 283 patients treated with tenecteplase after the switch. The door-to-needle times reduced from 61 to 53 minutes. The 3-month modified Rankin scale scores improved with tenecteplase switch, though not statistically significant. Numerically, the rate of symptomatic intracranial hemorrhage reduced, and deaths at 7 days also reduced with switching to tenecteplase. This is an experience from the Dell Medical School in Austin, Texas, that was presented at the International Stroke Conference and to be published eventually. These slides were given to me courtesy of Stephen Warwick. As you can see in these slides, they looked at the inter-hospital transfer times when they switched from alteplase and tenecteplase, and there was a reduction in the door-in, door-out times from 110 to 78 minutes, from the bolus to door-out from 52 to 40 minutes, and from the bolus to puncture time at the Comprehensive Stroke Center from 124 to 105 minutes. The rate of symptomatic intracranial hemorrhage, similar to the New Zealand experience, was numerically lower in the tenecteplase group, though this is not statistically significant. And then the discharge status, comparing the two groups, showed no significant difference in the two groups. However, numerically, there appeared to be an advantage in the tenecteplase group when looking at discharge to home, independent ambulation, or both outcomes as a combined status. And finally, this is the 90-day outcomes in the two groups. The modified Rankin scale score, 0 to 1, compared to the meta-analyses done previously, was similar, as was modified Rankin scale score 0 to 2. So mirroring what was observed in research trials. In terms of the guidelines for management, currently in the guidelines from 2019, the 2B recommendation is that it may be reasonable to choose tenecteplase as a single IV bolus of 0.25 milligram per kilogram, maximum 25 milligrams overall IV alteplase in patients without contraindications for IV fibrinolysis who are also eligible to undergo mechanical thrombectomy. And then secondly, they write that tenecteplase at 0.4 milligrams per kilogram single IV bolus has not been proven to be superior or non-inferior to alteplase, but might be considered as an alternative to alteplase with minor neurological impairment and no major intracranial occlusion. And this was based primarily on the NORTEST study of 1,100 patients. So in terms of current use of tenecteplase worldwide, it is widely used in Australia instead of alteplase, as you saw before. It is currently used in New Zealand primarily instead of alteplase. Multiple regions in Europe, Morocco has converted over to tenecteplase, UT Austin, Cedars-Sinai in the United States, Oregon Health Science Center, and within New England, Maine Medical Center and Bay State Medical Center have converted over to tenecteplase. And in research trials, the MOST trial, which is looking at eptifibatide and argatroban as add-ons to a thrombolytic agent in acute ischemic stroke, have allowed tenecteplase as the fibrinolytic drug in addition to alteplase. So in terms of getting institutional approval, this is the usual pathway. First going to the Pharmacy and Therapeutics Committee, which then reviews the application. The review then continues with other groups in the hospital who use lytics, such as the cardiology group, interventional radiology group, and pulmonary group. And as I mentioned earlier, pulmonary embolism does not have an evidence base for the use of alteplase, of tenecteplase in that condition. The safety committee then reviews it, and that was ultimately where we had to stop at UMass because there were concerns of confusing the drugs in the emergency department. And then ultimately, if it does go through that, it is reviewed by a legal department, and the whole process in general takes about 9 months. So in conclusion, tenecteplase is more fibrin specific than alteplase and appears to be more efficacious in large vessel occlusion. At this time, data from randomized trials have not definitively proven superiority or non-inferiority of tenecteplase over alteplase in non-LVO stroke, though no studies have shown that alteplase is superior to tenecteplase. Tenecteplase can be given as a single bolus in less than 2 minutes, typically in 10 seconds, potentially offering time savings in door-to-treatment times and transfers between hospitals. and the decision to transition to tenecteplase from alteplase should be considered in light of institutional concerns and competing needs. These are the references for this talk, and I'd be happy to take any questions.
Thank you so much for this very great update on most likely a drug that we will have to use at UMass, it looks like, and it has a lot of advantages. So I will open the forum for questions. I don't see anything in the chat. Anyone? Brian, on the twist, maybe I didn't capture exactly, on the twist study, was a placebo control, or was not the alteplase there? And that's why it was ethical to do that study if it was placebo.
Yes, a great question. There was no placebo. They were focusing on people who woke up from stroke, so those people would normally not get alteplase anyway. So these are people who went to sleep, woke up from stroke, and traditionally those patients are not eligible for treatment. And what they did was assign them either to usual care, which means, maybe get an aspirin or something like that, or whatever the physician chooses at the time, versus getting a lytic, in this case, tenecteplase, but they had to be within 4.5 hours of waking up from sleep. So those are people who traditionally don't get alteplase, and that's why they didn't have the alteplase comparison arm.
But I still don't understand, because they have this time frame that it's less than 4.5 hours, yes?
Right, but not from onset, from waking up. So the onset.
From waking up, okay, that's
right,
the clue, yeah.
Correct.
Okay. Dr. Alam, thank you for being with us. You have the, okay.
Thank you. Hi, Dr. Silver. Thanks for the talk. I was just curious to know whether, how the neuro-IR folks introduce intraarterial thrombolysis, whether there's any use for tenecteplase in that, or because I haven't seen anything like that out there. Just curious.
Yeah, that's a great question. I have not seen any studies using tenecteplase as the intraarterial lytic. Because they're directly in the vessel, they may figure that it doesn't matter as much in terms of having something that's more fibrin specific, although, as you saw, the half-lives are different. It's 5 minutes with alteplase, 20 minutes with tenecteplase, and they may be looking to try to get something that's more fast-acting rather than less fast-acting with intraarterial treatment. But I think that really does merit comparison in the study. But I would highlight that intraarterial lytic treatment has not actually been FDA approved, so technically that's an off-label treatment.
Brian, why tenecteplase is not indicated in pulmonary embolism? Was not studied, or it was not efficacious?
That's correct. It hasn't been studied. Okay. So it's just a lack of data as opposed to negative data. And that's really what's been the barrier here to switch over is all the other groups were fine with tenecteplase, but we would have to have two sets of lytics in the emergency department, and there was a concern about a potential mishap if the wrong drug was chosen. But I do think there's a workaround if the drugs are labeled appropriately, stroke TPA, PE TPA, and so on.
Thank you so much. I think we are a little bit behind with the time. And I have the pleasure to introduce Dr. Majaz Muniz talk. I'm just looking to see if Dr. Muniz is with us for questions at the end. If not, I don't see him. So I will ask.
I am.
You are?
Yeah.
Oh, good. Okay. For some reason, I didn't see her. I was ready to ask my colleague, Dr. Brian Silver, to help you, but that's okay. So it's my pleasure to have you in our program, and you can go ahead, Majaz. Thank you.
It's a recorded program.
Firstly, I'd like to thank all the organizers of the CME program for giving me the opportunity to speak about new advances in stroke prevention. We have had certain interesting advances over the years, which have allowed us to be more effective and safer, as well as improve outcomes in stroke prevention. Ischemic strokes are evenly divided between small vessel, cardioembolic, large vessel, and embolic strokes of undetermined source. It is interesting that all ischemic strokes seem to have a common strategy of prevention with their own specific subsets of interventions. For example, most patients would require antiplatelet drugs, high-dose statins, and optimization of their lifestyle and risk factors, including hypertension, hyperlipidemia, diabetes, and other risk factors. Then we come to the specific etiologies such as large artery atherosclerosis. If there's extracranial stenosis greater than 70%, which is symptomatic, then CEA or stent within 48 hours to 2 weeks is suggested, because that is the period where the highest risk of recurrent strokes exist. Asymptomatic stenosis does not require intervention unless there has been rapid progression despite best medical management. A randomized controlled trial, the CREST-2 trial, is testing this hypothesis, and we will see the results when we have them. For atrial fibrillation, anticoagulation with either warfarins or newer anticoagulants, and for other cardiac issues or heart alterations, other different strategies. Antiplatelet drugs in combination appear to have a synergistic effect and seem to have a better outcome of acute strokes. Now, does this apply to all ischemic strokes or not? There were 2 large trials that compared dual antiplatelet therapy, that is clopidogrel and aspirin, with clopidogrel alone in the MATCH trial or with aspirin alone in the CHARISMA trial, and both failed to show any efficacy over the monotherapy. However, in both these studies, the risk of symptomatic hemorrhages was doubled. So obviously not all ischemic strokes, but what subsets would benefit? Dual antiplatelet therapy with aspirin and clopidogrel in patients with minor strokes or TIAs, high-risk TIAs, was found to be superior than aspirin alone in a Chinese trial called the CHANCE trial. The combination given for 21 days after a minor stroke or TIAs reduced the risk of recurrent strokes by 32%, or absolute risk reduction was 3.5%. The trial was replicated in the United States and European countries, which is called the POINT trial, where again, the combination was tested against aspirin with very similar results, showing that aspirin and clopidogrel alone together were more effective than aspirin alone for the first 21 days. And after that, there was no added efficacy. This was very similar to the CHANCE trial. Furthermore, it also showed that the risk of bleeding increased subsequently with prolonged therapy. Based on these 2 studies, it was formulated that the dual antiplatelet therapy is appropriate for patients with high-risk TIAs or minor strokes for 21 days. Because of the risk of resistance with clopidogrel, another trial using ticagrelor was designed to test the hypothesis whether this drug, along with aspirin, was superior to aspirin alone in the same TIA and minor stroke population. The only difference was that the stroke population was had an NIH stroke scale of 0 to 5, not 0 to 3, so somewhat higher level of severity. What the trial showed was, again, that the combination was superior to aspirin alone, and that symptomatic bleeding was similar in the two groups. In subgroup analysis, patients with atherosclerosis did better than those without, and the benefits extended across the entire range of high-risk TIAs and ischemic strokes from NIH stroke scale of 0 to 5. The CHANCE-2 trial is a further advancement on understanding how dual antibiotic therapy should be applied. There's a large section of the Chinese population especially, and maybe others, who carry the CPY2C19 loss of function allele. And in the CHANCE trial subgroup analysis, it was shown that these are the patients who did not benefit from the combination therapy of aspirin and clopidogrel. Based on this, the CHANCE-2 trial was designed and tested whether ticagrelor plus aspirin would overcome this problem of clopidogrel and aspirin in the initial management of these high-risk TIAs of strokes. And the trial did show that ticagrelor and aspirin is superior to clopidogrel and aspirin in this group of patients. The risk of bleeding was somewhat higher in the ticagrelor group, but risk of symptomatic bleeding was not higher. And there was a clear advantage. This obviously leads to the question that should we test patients before initiating these combination therapies for resistance, and then appropriately time them or triage them to the clopidogrel aspirin group or ticagrelor aspirin group? A difficult question to understand and manage because these tests are not available to us immediately, but they're getting better, and soon we should have that answer on how to triage these patients. So management of TIAs should always include a quick appraisal of to see if they have ongoing atrial fibrillation, meaning which is monitoring for the first 24 hours, looking for carotid disease that could be treated urgently, and if those are not present, then treat them with high-dose statins, antiplatelet drugs, and perhaps dual antibiotic therapy for 3 weeks, unless they have symptomatic intracranial arterial stenosis, in which case the duration extends to 3 months. In the Oxfordshire study, it was shown clearly that early intervention of this type reduced the risk of recurrent strokes by 80%, which is very impressive and points to the urgency of management. What about statins? Statins have been the universal wonder drug and have shown in primary prevention trials to have a 24 to 30% risk reduction in subsequent events. In the heart protection study, there was a 24% reduction in all vascular events and 17% reduction in vascular deaths, which was highly significant. This was with simvastatin 40 milligrams daily. There were no significant adverse events such as rhabdomyolysis or bleeding or cancer, some of the things that were thought to be related to statins early on. SPARKLE was a follow-up study after the heart protection study, and basically looked at patients with ischemic strokes without any cardiovascular disease to see if there was indeed a difference between using high-dose statins or not. In this trial, atorvastatin 80 milligrams versus placebo was tested, and found that there was a significant advantage of the high-dose statins with a 2% absolute risk reduction and an 18% relative risk reduction in preventing recurrent strokes. Following this, there was a trial of patients who had normal lipidemia, but elevated markers of inflammation, such as sensitive CRP. These patients were treated with rosuvastatin 20 milligrams daily and reduced both the risk of stroke and MI by 44%. So CRP, even patients who have normal lipid levels should be treated with high intensity statins to try and reduce the early inflammatory effects that are associated with the stroke. Statins improve outcome of stroke, as we've shown early on, and subsequently reproduced by Dr. Flint's group, who also showed that the earliest intervention, the better the outcomes. So in the first 72 hours, the outcomes are really great, and after that, the effects start to be less noticeable. What should be the target for LDL cholesterol? The Treat Stroke to Target study determined, based on intense lowering of LDL cholesterol to less than 70 or more liberal lowering to 90 to 110 milligrams per deciliter, that with the intense lowering group, there was a 12% lower risk of recurrent ischemic events. This is in keeping with the heart protection study, which also showed similar results. This less 70 milligrams per deciliter was the sweet spot, and we should keep LDL cholesterol below 70 milligrams per deciliter in all groups. In specifically cardioembolic strokes, with no other known factors, again, statins were found to be superior in preventing major vascular events, vascular death, and all cause death. So the argument that statins should not be given in cardioembolic strokes, in my opinion, is not justified. All acute strokes should be treated with high-dose statins. Statins are also protective against intracerebral hemorrhage. At least that is the initial data. But there's an ongoing trial to test this hypothesis. The trial's name is STATIN, where patients are randomized to continuing statins after intracerebral hemorrhage or not continuing statins. Results will be interesting and will pave the way for further use of statins in this group as well. The pleiotropic effects of statins are far more important than the lipid-lowering effects of statins alone in terms of improving acute stroke outcome, which is why they should work for all subgroups, including cardioembolic strokes. The lipid-lowering effect is important for secondary prevention in the long term is used for that purpose. But LDL has never been found to be a predictor of outcome and should not be used to start or stop statins in acute ischemic stroke. In cases where patients are refractory to statins or cannot tolerate statins, alternatives are PCSK9 inhibitors, which act by a different mechanism, but have the same pleiotropic effect as statins and are cardioprotective as well. In patients refractory to statins, adding the PCSK9 inhibitors reduces the LDL levels by another 50%. And they can be used independently to lower LDL cholesterol in patients that are sensitive or have shown adverse effects to statins. 4 new oral anticoagulants have been available for the last 5 years. 2 of these are superior to warfarin and 2 are non-inferior to warfarin. Advantage: they have no ramping effect. They're therapeutic immediately on day 1. They're also drugs that do not require any monitoring. They are associated with less degree of symptomatic intra or extracranial hemorrhage, and do not interfere with lifestyle, such as limitation of kind of foods that you have to curtail with warfarin, specifically vitamin K rich food. Of these 4 drugs, apixaban seems to be the best in terms of reducing the risk of symptomatic bleeding and also being superior to warfarin in terms of efficacy. In the Evros trial, there was no increased risk of bleeding, symptomatic bleeding, compared to aspirin. There are some specific situations that require specific treatments that are different from our usual stroke prevention management. Cerebral amyloid angiopathy and atrial fibrillation often coexist in this older age group and pose the challenge of what anticoagulation therapy should be instituted. The risk of bleeding with anticoagulation is 9 times higher in patients with cerebral amyloid angiopathy as compared to those without. Alternatives to the treatment are to use devices instead of anticoagulation. And there are 2 such devices. One is the Watchman device and this other is the Amplitzer device. Both are designed to occlude the left atrial appendage. And in trials have been shown to have the same efficacy in the short term in reducing risk of recurrent ischemic strokes. PFO closure is a tricky question and should be only attempted in patients who are young, who have no other risk factors, and who do not have any hypercoagulable state or atrial fibrillation, which should be they should be monitored for atrial fibrillation before PFO closure is considered. It should be a joint decision between the neurologist and cardiologist when or who to close. Embolic strokes of undetermined source do not have a real treatment at this point, but 2 trials trying anticoagulation, the Navigate and Respect, were negative, and a 3rd ongoing trial, the Cardia trial, is ongoing, testing apixaban against aspirin in this group. Embolic strokes of undetermined source are what they are. We don't know what the cause is. We think they're embolic, but we're not sure. So we treat them with conventional therapy, and we should monitor them with implantable loop recorders in the long term to see if they indeed turn out to be atrial fibrillation, or as others think, atrial arteriopathy as the cause. Maybe there are other causes that we have not explored yet. How long should patients be monitored, and how should they be monitored for atrial fibrillation? The conventional methods obviously are very inferior, and implantable loop recorders show a far superior rate of pickup of atrial fibrillation, as well as help improve outcome by timely anticoagulation. Sleep apnea has been known to cause atrial fibrillation, hypertension, increase the risk of myocardial infarction and ischemic strokes. It has been overlooked for a long time in stroke prevention management, and data now emerges that shows that patients who are treated, identified and treated early with CPAP or other interventional strategies would have a reduced risk of having recurrent strokes or myocardial infarction or other cerebrovascular disease deaths. Finally, more recent studies have shown from Canada that post-stroke mortality is reduced by 50% with continued moderate severe exercise. And do not forget the Mediterranean DASH diets improve or reduce the risk of having recurrent strokes. Thank you.
Thank you so much, Mojaz, for this nice review of prevention of the stroke. Any questions? I'm looking for somebody with a hand up, but
there's a question here.
Yep. Oh, Dr. Alam. Yep. Thank you.
Thank you, Dr. Muniz. I just had a question regarding a particular scenario. If somebody were to face in the clinic, you know, like a 60-plus-year-old person comes into the clinic and they're complaining that they had, you know, transient weakness or they had transient dysarthria, something of the sort that was, you know, maybe more than 2 weeks ago, but like within a month. And you suspect that they're having a TIA. Especially if it's like coming from, you know, the left side, excuse me. So they're having some kind of weakness on the right or difficulty speaking. You were to get an MRI and it showed that the patient had a stroke. I know it's out of that 2 weeks, but how do you usually approach in terms of, do you still kind of have them be evaluated or seen by someone to address their carotid arteries in case they had, you know, critical stenosis? Or I'm just curious, I've run into that a couple of times before. So I was just curious, how do you approach a patient who has critical sort of carotid stenosis, sort of symptomatic because they had a stroke in that particular area, but they're just out of that 2-week window?
That's a good question. And no, I think the answer really is that the 2-week window is only based on the fact that that is the time when you have the maximum risk of recurrence. But that does not mean that beyond that, the risk of recurrence becomes zero. So yes, I would definitely get them evaluated. And if they have significant symptomatic stenosis, then it should be treated, whether with CA or with stent, depending on what the, you know, other comorbid conditions are, etc.
Dr. Winkenfeld has a question. Go ahead, Ari.
Hi, very good talk. If statins are effective in the short term, independent of whether someone has already or phytostatins are effective in the short term, independent of whether someone is already in or close to the lipid lowering target, when does short term become long term from a practical standpoint? If you have a patient who's there. You know, we started high dose statins. Their LDL comes back at 67 and they're ready to leave the hospital. How long do we leave them on a statin for?
So typically, there's been no cutoff answers, but the approach is to leave them on for at least 3 months and reassess it at that point. I wouldn't worry too much about the LDL levels, because LDL levels up to 30 have been shown not to be associated with any increased risk of bleeding.
It wasn't the LDL level, it was more the patient who is hesitant to be on medications and wants to know, how long do I have to be on it?
Right. So the answer is really that you should be on it forever, but in lower doses.
Okay. Thank you.
Yeah.
Anyone else has a question for Dr. Muniz? I have a question for you, Majaz. How often have you prescribed the PCSK, or do you refer somebody to the metabolism clinic if you need to use one of the monoclonal antibody?
So I have prescribed that about 4 or 5 times so far, not very often. And I have referred some of them to the lipid clinic only because Dr. O'Keen has a mechanism set up for getting the drug approved much faster. As I said before, the drug is highly effective and convenient, and reduces LDL cholesterol by 50%. And it's also an add-on drug for people who are refractory to statins.
Thank you. Thank you for this answer.
Well, thank you.
Any other questions? We are pretty good on time, and we have Dr. Puri with us. And thank you for being with us today. And we have his talk, and we can go ahead. Dr. Puri is a professor of radiology, neurosurgery, and neurology, and the director of the neurointerventional radiology program here. He's not part of the neurology, department as a full-time. He's definitely an affiliate with our department and a very nice collaborator.
Hey, good afternoon, everyone. This is Dr. Puri. I'm going to talk about stroke thrombectomy trials, current evidence, and future implications. These are my disclosures. Stroke remains the #1 cause of disability in our country. It used to be the #3 cause of death. Now we have done a good job with that, and it's now a #5 cause of death. Every 40 seconds, somebody has a stroke in our country. About 87% of those strokes are ischemic from lack of blood supply, of which about 30 to 50% can be due to large vessel occlusions. Now, these numbers are a little bit antiquated from the stroke burden in terms of the cost, but these are well north of $200 billion in terms of direct and indirect cost to patient and their families, and the lack of work supply afterwards. This is a very common slide. Everybody knows about that, but it kind of does drive home the point, like every minute we're losing about 2 million neurons. That amounts to each hour of stroke where we have a large vessel occlusion. The number of years our brain ages is about 3.6 years. So those patients lying in the hallways in the ED who are not complaining because of lack of pain or no broken bones, they are the most dangerous kind of in terms of losing functional life. What is the natural history of these kind of things? If you look at just the different locations in an MCA, mortality could be anywhere from 30 to 35%. In an ICT occlusion, it's about 50%, like half of the patients with an ICT occlusion might die. We're not talking about morbidity, we're talking about mortality in these patients. Bachelors, all bets are off. These are almost up 90% mortality, especially depending on the location in the bachelor. These are what we mean by large vessel occlusions. When we come up from the carotid, if there is a complete carotid occlusion from the neck up there, it's the total IC occlusion. Right up where the carotid bifurcates and goes medially into the anterior cerebral artery and laterally into the middle cerebral artery, that junction is, which looks like the letter T, is called a T occlusion. The part of the MCA before the bifurcation is called the M1, and after the bifurcation called M2. There is still some debate on the physiological M2, surgical M2, and the radiological M2, but this is the bifurcation. Bifurcation is what we use to define M2s. By NIH, which is very commonly used which is used actually in every stroke patients, you have an NIH of more than 12, equal to 12, you have more than a 90% positive predictive value that this patient has a large vessel occlusion. Depending on other studies, you have NIH of 10 or more, you have almost a 85% chance that the patient we are dealing with has a large vessel occlusion. These are the current ways how we treat a large vessel occlusion. Medical management still remains the cornerstone of every stroke treatment, be it the patient gets TPA, not TPA, thrombectomy, not thrombectomy, but the medical management still remains the cornerstone. FDA approved IVTPA back in the late 90s, actually 1994, where it remains the gold standard for any stroke, ischemic stroke. Then came the bridging therapy to endovascular clot retrieval, and then patients who are ineligible for IVTPA can go for TPA, go for thrombectomy in addition to TPA or directly for endovascular clot retrieval. This was the first approved device for clot retrieval back in 2004, where this looks like almost like a wine corkscrew. This was called the Mercy retrieval device, where we used to put a microcatheter distal to the clot over a small micro wire, deploy this corkscrew-like device in the level of the clot, inflate the balloon in the carotid artery in the cervical region, and then pull this thing out. This is how it looked. My first 44 or 45 cases of acute stroke thrombectomy were with this device, a very archaic device, which is probably not in use anymore. But this is what it looked like, where you had a young patient with a right distal M1, we could see by the clot, that corkscrew-like device, and when you pull it out, the clot comes out and you have the MCA open over there. Now, based on these older devices, you might know there were some trials done, like IMS3 is one of the ones which is very commonly used at that time. The problem with most of these therapies were we did not have very good recanalization rates. Like in this IMS3, which showed futility of endovascular clot retrieval, there was IA lytics, Mercy, ultrasound use, some other devices use. The stent retrievers, which we normally use now, were used in a minuscule of those cases for IMS3 and MRSQ, which again led to lower TQ2B recanalization rates, which then led to not good clinical outcomes or clinical outcomes equivalent to IVTPA. The turning point for stroke thrombectomy was when stent retrievers came. This is almost like a very fine mesh, which is held down by a wire, and the wire does not detach. And then you basically let the clot integrate with the stent and pull the entire thing out. This is a small cartoon of where you have an MCA occlusion. You pass your wire with it, with your own micro wire, you cross it, then pull your micro wire out, and then through this hollow plastic tube called the microcatheter, you bring in your stent retriever. The stent retriever, as soon as you deploy it, it creates a channel for the blood to go in, something called as ischemic holiday. So from that channel, at least there's some blood going. Usually anywhere from 3 to 5 minutes, we wait at that time and let the clot integrate with the stent retriever, get caught in the struts over there. Once it has done that, then we inflate the balloon in the neck and pull this entire assembly in toto as a single assembly. That's the balloon inflating in the neck. The rationale for inflating the balloon in the neck is so that none of the clot breaks up while it's going through all these turns and tortuosities in the ICA terminus siphon all the way to the cervical ICA. And then once you've done that, you clean up the carotid for all the microemboli still there, and then deflate the balloon and do a run after that. So this is what the stent retrievers led to. Multiple randomized controlled trials were done, which were published. 4 of them were published actually in the same NEGM journal issue in February 2015. MR Clean was in Netherlands. Escape was in Canada mostly. Extend IA was in Australia. Swift Prime was in US. And UMass was the only site in Massachusetts which enrolled in this trial. If you look at the median baseline NIH, very similar. And in most of these trials, it was the stent retrievers, if not exclusively, the majority of the cases where the stent retriever used. And that's what it led to. Once we started using stent retrievers in these cases, the recanalization rates, the TT2Bs and 3s for the interventional arm rose, and that led to the better, good clinical outcomes as compared to the control arm, and as high as 71% in Extend IA. So these are again showing that first we have to recanalize. If you are unable to recanalize, you're not going to get good outcome, and the good outcomes are directly related to the revascularization or recanalization rates. And these are the modified Rankin. You can see as compared to control, every one of them showed statistical significance in terms of at least the ones which were done in US was Swift Prime, and MR Clean done in Netherlands, and Escape in Canada. This is an example of an MC occlusion. You can see there is a cutoff right in the M1. And this is what I meant by deploying the stent retriever. You can see a small thin channel, which actually shows that the contrast is not going, which means the blood is now going into the MCA territory. You let it wait for a few minutes and then you pull it out. And then this is what you see. That's the entire MCA, which was penumbric or was not getting the supply. And we have hundreds and thousands of these cases where you can see the big chunks of clot coming out with the stent retriever. This is another case of a top of the basilar, which is one of the most critical scenarios we'll see as a thrombectomy patient. This is the top of the basilar gone with both of the PCAs gone. And there's the stent retriever one time in the right PCA and one time in the left PCA, and the entire vessel is open over there. So this initial 5 randomized controlled trials, which were published in 2015, led to AHA and ASA revising their guidelines and calling us a level 1A evidence for LVOs within the first 6 hours. That's what most of the trials used. We look at the pre-morbid, pre-stroke modified Rankin. We're looking for young, healthy individuals, age above 18, occlusion of the ICA or the MCA, NIH of equal to or more than 6, and aspect score of 6 or more. A little bit on aspects. This is what we meant by aspect. This is a non-contrast. If you look at the MCA and the non-contrast, it has given it 10 different points, and you deduct point for each part of that MCA or deep gray matter where you have hypodensity on the non-contrast CAT scan. An MCA which has only 3 areas of hypodensity will have an aspects of 7 that way. Now, till this time, we were in the first 6 hours, which had excluded most wake-up strokes, patients after 6 hours. So then came diffuse 3, which was actually terminated because of the efficacy, which triaged patients on the MR and the MR perfusion characteristics. They looked at 38 US centers, of which 182 patients were randomized before the trial was stopped because of the efficacy, in equal 92 in the endovascular arm and 90 in the medical therapy arm. What it led to was a good modified Rankin of 0 to 2 in 45% of the cases where the thrombectomy was done, and only 17% of the cases where it was not done. So that's why it was stopped prematurely, which led to the conclusion that in eligible patients which are chosen based on imaging characteristics, now you can extend the thrombectomy windows from 6 to 16 hours, so which opens up from 0 to 16 hours in these cases. Then came the DAWN trial, which looked at more day-to-day how we use CTs, like this use CT perfusion as compared to MR, which is less likely to be used for triaging of acute stroke patients. Not everybody is same. It's our stroke volume and our penumbra is a function of our collaterals. And that's what it looked like with DAWN, that we wanted to look at the collaterals and how collaterals can sometimes sustain patients beyond the 6 hours also. So this looks from 6 to 24 hours. This was one-to-one randomization of medical management plus medical stent retriever versus only medical management. The plan was to enroll about 500 patients, about 215 each arm. But again, it was stopped at 206 because of the efficacy, which was published in NEGM again from 6 to 24 hours. That's what it looked like. About 50% of the patients who got thrombectomy in the 6 to 24 hour window were having good outcomes versus only 13% in the control arm. More secondary analysis looked like almost 2.5 times more patients were like to get a dramatic decrease in NIH of more than equal to 10 in the thrombectomy arm as compared to the control arm. And the revascularization, of course, was higher in the thrombectomy arm, almost up to 80% versus 40% in the medical management arm, which led to expanding of the time window from 6 to 24 hours in LVO in the anterior circulation based on CT perfusion, DWI, or MR perfusion, needing extra advanced imaging. Now, recently, if you followed the recent publication of the CLEAR study, it actually brings this matter up for debate, where they looked at the CT perfusion and MR and looked at and it said that CT was actually as good as CT perfusion in triaging these patients in the 6 to 24 hours also. Now, how powerful is this treatment of stroke thrombectomy? We know antibiotics in septic shock is like a given, it's commonsensical. That's how strong this treatment is in achieving functional independence for these patients at 90 days. The need to treat is as low, between 3 to 7 to treat these patients to get functional independence, as opposed to STEMI, where you have to treat 45 to 91 more patients to get one more additional short-term benefit in short-term mortality. In terms of healthy time lost, the Hermes meta-analysis also looked at that from the 7 randomized controlled trial. What it said was once we have in hospital, once the patient comes into our door where there is an LV and LVO, each 10-minute of delay, we are losing about 2 months of healthy life loss. That's amounts to 1 second of delay, about 2.5 hours of healthy life is lost. Now, moving on to future implications, we are looking at large core. What about aspects? And this is another refresher. What we're talking about aspects is what about patients who have a large stroke? Remember, in the initial trials, it was 6 or above. What happens if the aspects is 0 to 5, which makes the patient already has stroke in 5 of those 10 or more than 5 of those? What about the lesion volume being above 50, above 70, above 100? So there were some publications which have come out of this. One is called the SELECT study, which looked at a small number of patients, was published in JAMA Neurology a couple of years ago. They looked at about 105 patients, of which patients who were treated with thrombectomy, about 1/3 of these patients achieved functional independence, as opposed to only 14% in the patients who did not get thrombectomy. Now, we are talking about patients with an LVO with a large stroke, and we are talking about 1/3 of them getting functional independence. So in the endovascular arm, there was better functional outcomes, less infarct growth, and smaller final infarct volume as compared to the control arm. Now, mind you, none of those patients who had a stroke more than 100 cubic centimeter had any favorable outcome. So at this point of time, it looks like there might be a tipping point of about 100 cc. Anything above that on the initial CAT scan or any advanced imaging, the patients are probably not likely to get any benefit, or for that matter, might actually have more risk as opposed to patients who have less than 100 cc's of contrast of stroke on the initial scan. Just looking at the multi-center data from Europe, also from multiple institutions, aspects 0 to 5 with an LVO on the anterior circulation, it does matter whether you open the vessel or not. In this case, if you're looking at modified rank in 0 to 3, once we open the vessels, about 50% of them can achieve functional independence versus 13.9 or 14% in the patients where you do not open the vessel. So mind you, there was no control group, and this is a retrospective case series and analysis from multiple institutions in Europe. Ongoing trials which are going on for large stroke, one is called Tesla. UMass is a site for Tesla where we're looking for aspects 2 to 5 and within 24 hours. Tension is a trial which is coming out of Europe, and LAST is another trial which is happening within first 7 hours of aspect 0 to 5, which is going to be completed next year. Low NIH, we know for the guidelines, it's 6 or above. Below that, we have actually randomized controlled trials going on. One is called Endolo. UMass is a site for Endolo where we're looking at patients with IC and M1 occlusions with an NIH of 0 to 5, which are getting randomized to medical management versus thrombectomy. And similarly, MOST is also looking at patients with medical management versus thrombectomy in the NIH 0 to 5 window. And there is a rescue arm also. I think I have used up all my time. Thank you so much.
Thank you so much, Ajit. Any questions? We can have one question because of the time constraint.
This is Niels. I will be quick then.
Yes, thank you, Niels.
Ajit, wonderful. I mean, of course, we're all big believers in this, and we've seen the miracles that your team can do with these patients who are so severely affected. Where do you see the future for this? Where are we going with this? And maybe specifically comment on adjunct therapies with neuroprotectants or so.
I think that's a wonderful question, Nils. I think we have reached the maximum what we can do technologically with catheters and devices. I think the next big thing is pre-hospital. How do we freeze that penumbra? How do we decrease the neurotoxicity of the ischemic brain? And more specifically, like you talked about neuroprotection. Like 15, 20 years ago, when all the neuroprotection trials were done and we had a egg on our face, the reason why at that time we did not work was we were not able to recanalize those things, the vessels specifically. Now, we are able to recanalize these vessels up to 95, 98% of the times. Now, neuroprotection is making a big comeback. You already have Escape published, which showed that in the non-TPA arm, there was benefit in the neuroprotection. Escape NA Next has already started. UMass is a site. These are for patients who are ineligible for TPA. That's where it is. 2 more phase 2 trials are starting, which are looking at, one of the drugs is an actual analog of insulin, which is looking at decreasing the core stroke in 0 to 5 aspects and showing better outcomes in that. So I think what you will see in the next year or 2 are multiple trials with neuroprotection plus thrombectomy or any therapy which will open up the vessel. So that's where I think our field is heading now.
Thank you so much, Ajit. This was a great review of what it's done in the literature and partially what you guys are doing, because you are doing much more than what is done anywhere close by. So for our patients, and we are so happy to see the review of the studies that you guys do, because it's important for everyone to try to refer the patients to you to be included. So
thank you for having me.
Thank you. Now we move on. We are not taking a break because, I think everyone wants to maybe have a dinner and to not, prolong more the stay in front of the computer. And I have a great pleasure to invite Felicia Chu, who is an assistant professor in our department in the Division of Epileptology, and she is actually the director of that. And Felicia, you are on.
I'm here.
Good, thank you.
Hi, my name is Felicia Chu. I'm an adult epileptologist here at UMass. I'll be talking today about seizure detection devices. I have no disclosures. Many of the devices that we'll be discussing are not FDA approved. Goals and objectives will review ongoing developments and availability of wearable devices for automated seizure detection for patients with epilepsy in the outpatient ambulatory settings. Hopefully, this will provide healthcare personnel who are working with patients with epilepsy some information and guidance on how to counsel their patients on the use of this technology. Wearable devices are becoming widely used, and this trend has reached healthcare applications, including epilepsy. There are hundreds of wearable devices on the market that measure health parameters and biosignals, and many of them make unsubstantiated claims of detecting epileptic seizures. We'll look at the methods of detection and efficacy and discuss clinically validated wearable devices for automated detection of convulsive seizures. Finally, we'll review evidence-based guidelines for clinical implementation of automated seizure detection using wearable devices, as there is potential for wearable seizure detection devices to improve clinical care and quality of life for our patients. Why do we need improved seizure detection? Approximately 1/3 of patients with epilepsy are not seizure-free, in spite of adequate treatment. Clinicians manage patients with epilepsy and conduct drug trials gauging the efficacy of interventions based on subjective seizure count. Patient and caregivers' self-reporting of seizures can be unreliable. People with epilepsy may be partly or fully amnestic to seizures. Individuals may be unable to provide an accurate account of seizure occurrence and severity. And witness accounts of epileptic and behavioral events are often inconsistent. Studies in video EEG monitoring units demonstrate that 47 to 63% of seizures remain unrecognized by patients, and this is even higher at 86% for nocturnal seizures. Generalized tonic-clonic seizures, including focal to bilateral tonic-clonic seizures, can lead to injuries and is the main risk factor for sudden unexpected death in epilepsy, SUDEP, especially in unattended patients during nighttime hours. Each year, about a quarter of patients with generalized tonic-clonic seizures experience at least one serious injury related to the GTC, causing disability or requiring hospitalization or surgical intervention. SUDEP is defined as death in people with epilepsy not caused by injury, drowning, or other known causes, and is thought to be associated with post-ictal cardiopulmonary arrest. The rate of SUDEP is significantly higher in patients with more than 3 tonic-clonic seizures annually and nocturnal convulsions. Therefore, automated detection of GTCs may help decrease morbidity and mortality associated with seizures by alerting caregivers to ensure prompt attention. Patients with generalized seizures and those with focal impaired aware seizures are not able to call out for help during seizures. Unpredictability of seizures contributes to social isolation and decreased quality of life. Furthermore, this unpredictability of seizure occurrence is distressing for patients and for caregivers, and detection provides an element of patient empowerment and opportunity for intervention. The disparity in perceived versus objective seizure frequency and the need to detect convulsive seizures for safety purposes makes wearable seizure detection devices a possible solution to these problems. Currently, there are 5 modalities utilized for seizure detection, and they may be used in combination or in isolation. Most of these have a high sensitivity for convulsive seizures, but a low specificity due to artifacts. Additionally, they are poor at identifying non-convulsive seizures. These include electroencephalography or EEG, heart rate detection, electrodermal activity, motion detection, and electromyography or EMG. EEG remains the gold standard for seizure detection. Several EEG-based seizure detection devices have been developed that come in the form of either a headset or cap-like configuration and substitute for standard EEGs that use electrodes glued to the head. Although these headsets or cap-like configurations are often utilized in the laboratory only. Newer EEG devices allow for a longer recording times and also have a reduced number of electrodes. For example, the 2-electrode 1-channel scalp patch shown in the picture on the right can be applied for up to 1 week. Additionally, there's a 4-electrode 2-channel subcutaneous device that can be implanted and remain up to 30 days of recording. Limitations include the requirement of prior knowledge of the seizure onset zone. Given the limited number of electrodes, it needs to be near or close to this, as well as the potential for artifact. There's false positive seizure detections. There's a necessity for the clinician to interpret the findings of the recordings. And the device can be cumbersome, have short battery life, be costly, and furthermore is not FDA approved for treatment or diagnosis. Autonomic effects of seizures are well known, and a recent meta-analysis of heart rate changes before, during, and after seizures found 64% of generalized seizures and 71% of focal seizures were associated with changes in heart rate. One application of this is in the newer models of the implanted vagus nerve stimulators depicted on the upper right-hand corner. This vagus nerve stimulator will monitor heart rate and deliver an electrical stimulation to the vagus nerve when the heart rate increases by 20 to 70% of the baseline within 10 seconds, compared with the average over the previous 3 minutes. In a 2015 study evaluating efficacy in detecting and treating seizures associated with changes in heart rate, 43% of seizures were associated with greater than 20% change from baseline heart rate. The frequency of closed-loop VNS activation could provide an estimate for seizure occurrence, although VNS is not tested or approved for that purpose. Other examples of heart rate detectors include a watch-like device that detects changes in heart rate and sends a Bluetooth signal to the designated receiving device when heart rate falls below a specific threshold, hopefully decreasing the rate of SUDEP. Of course, the limitations would include the lack of detection of seizures without heart rate changes, or false positives associated with other causes of elevated heart rate, such as exertion. In addition to heart rate changes during a generalized or focal seizure, autonomic nervous system involvement may result in sweat gland activation or electrodermal activity. In 2010, a study looked at 7 patients with epilepsy evaluating EDA during seizures, 9 convulsive seizures and 4 focal seizures, with a watch-like device that detects changes in skin conductance of electricity. There was a sharp increase in EDA noted at the onset of all the seizures, with the most profound increase in convulsive seizures. This EDA detector was combined with movement detection to create a device that is now FDA approved for the purposes of detecting generalized tonic-clonic seizures. The device will also send an alert to the designated responder's phone. A 2015 study evaluated sensitivity and specificity looking at 69 patients with epilepsy. EDA and movement detection in the Embrace wristband yielded a sensitivity of 95% with a false positive rate of 0.2 number of false alarms per 24 hours related to repetitive movements. Motor seizures, and in particular generalized tonic-clonic seizure movement, can be detected using actigraphy or accelerometry. An example would be a wearable accelerometer detecting changes in velocity trajectory of movement. In one multi-center trial, 73 patients were monitored and 87% of seizures were detected, with a false positive rate of 0.2 detections daily related to repetitive movements like teeth brushing. Missed events were due to restricted movement of the limb wearing the device. Another device was studied in 2013 and evaluated detection of convulsions with a motion detector under the mattress, primarily for seizures during sleep. 51 patients were studied and 88% of the convulsions were detected. There were 93 other events that were missed, but these were often characterized by more subtle movements. 21 false alarms were related to repetitive movements. There has been some development of video-based devices with algorithms to detect movement consistent with convulsive seizures as well. Limitations of all the motion detectors include a high false positive rate associated with deliberate movements and also missed events. Surface electromyography, or EMG, is also been used for seizure detection. In a study in 2018, they evaluated an EMG-based device in 71 patients with a history of generalized tonic-clonic seizures over several days, wearing a surface EMG device on their biceps muscle. 32 GTCs were captured from 20 different patients, with 30 of those being detected by the device, with a sensitivity of 93% and a false detection rate of 0.67 detections per day, with the most 68% occurring during purposeful exercise. Limitations included a high sensitivity for convulsive and tonic seizures, but false detection still occurred, triggered by common tasks such as physical exertion. Currently, there are 2 wearable sensors approved by the FDA and the European Union for detecting convulsive seizures. The first is a wrist-worn smartwatch, the Empatica Embrace, that includes the combination of accelerometry and EDA for seizure detection. It has a Bluetooth link to a subject's smartphone where an app will then issue an alert to caregivers if seizure occurs. The second device is the Brain Sentinel, that is an adhesive patch affixed to the subject's bicep and identifies changes in EMG to detect convulsions. It too has a cloud-based data platform that can send caregivers alerts when a seizure is detected. Overall, limitations of seizure detection devices include a consistently significant false positive rate. There is a financial burden. Oftentimes these devices are not covered by insurance or only partially covered. These devices will miss subtle seizure manifestations associated with focal seizures. And most commercially available sensors do not have regulatory approval for use in epilepsy, including those functioning on smartwatches with accelerometry and photoplasmography sensors. So the International League Against Epilepsy and the International Federation of Clinical Neurophysiology published clinical practice guidelines earlier this year to provide clinicians guidance on utilization or recommendations for patient use of wearable devices for automated seizure detection. And this is what they put out. Wearable devices are effective for accurate detection of generalized tonic-clonic seizures and focal to bilateral tonic-clonic seizures. However, it is uncertain whether the detection alarms result in meaningful clinical outcomes for patients until further research is completed. They do recommend, however, wearable devices for detection of tonic-clonic seizures, especially in patients who are unsupervised overnight. Recommendations for improvement overall with technology include increased performance of wearable devices and detection algorithms for higher sensitivity and lower false detection rate, especially with seizures without generalized convulsion. Decrease the false alarm rate, allowing objective documentation of seizure frequency. Conduct properly designed clinical validation studies. Demonstrate whether automated seizure detection leads to meaningful clinical outcomes such as decreased morbidity and mortality associated with seizures, objective seizure quantification, and improved quality of life. In-field phase 4 studies are needed to provide a more accurate estimation of the false alarm rate. Similarly, costs, patients' preferences, and perspectives should be considered in the evaluation of the impact of this technology. Thank you for your attention today.
Thank you so much, Felicia, for this nice overview of a great, addition to our medication for those patients who are sometimes not even knowing, like you said, unsupervised. I open for discussion. We have a couple minutes if you guys want to ask any question. Go ahead. I don't see any hand up right now.
Sure, I can ask a question.
Yes, please go ahead.
Felicia, I was just wondering whether any of these devices, at least in your research, can differentiate pseudo seizures or non-epileptic spells from actual epileptic seizures or GTCs.
This is a tough one, Nabil. Okay. I don't know. I think Felicia, you are mute.
Maybe there's some technique.
Maybe she lost the ability to turn on the mic. I'm sorry, Nabil.
No, it's okay.
I don't think I have the answer to this question, but I can give you my best guess that it's hard. Very hard. Okay.
Thank you for trying.
So I think at this moment we can move forward with the talk of Dr. Kyle Rossi, who is an assistant professor of neurology in the epileptology, in the epilepsy division. And I think he is going to have a live presentation. So are you able to share the screen right now, Kyle?
Yes, I actually.
Are you?
Yes, I actually recorded it, so I'm going to hit play right now.
Okay. You go ahead with whatever you
enjoy.
Thank you.
Hi, my name is Kyle Rossi, and my goal today was to give you an interesting but clinically relevant update on EEG. The first EEGs were recorded by Hans Berger in the 1920s, and Gibbs and his colleagues first described epileptiform discharges in 1935. These are markers of increased seizure risk and can take the form of sharp waves, spikes, and spike wave discharges. In that same article, they described and documented the first waking patterns, sleep patterns, true seizures, and epileptiform discharges, which they amusingly called larval seizures, and coma patterns. From there, EEG was introduced into clinical practice. Digital EEG was introduced in the 1990s. Today, modern server, broadband, and network technology allow for high volume continuous EEG monitoring. Just a brief word on the neurophysiology. With EEG, we're measuring summated excitatory and inhibitory postsynaptic potentials from populations of cortical neurons that make up the normal rhythms of the brain, as well as various pathologic entities. We're measuring only the most superficial layers of cortex, and in total, we're only sampling about 1/3 of the entire cortex. EEG has a much higher sensitivity for radially oriented neurons, and significant natural frequency filtering occurs due to layers of tissue, bone, and fluid between the brain and the electrode. A commonly tested fact during an epilepsy fellowship is that 6 square centimeters must be involved in a spike discharge for the spike to be seen on a scalp EEG recording. And that same principle has even been extrapolated to seizures, such that not all electrographic seizures are seen on scalp EEG, especially those with limited clinical symptoms such as focal motor seizures. If we take a look at all available functional neuroimaging and we plot spatial resolution on the y-axis, temporal resolution on the x-axis, and invisivity on a z-axis from light to dark, we see that the relative strengths of EEG are high temporal resolution and low invasivity, but it has significantly lower spatial resolution than most imaging modalities. If we compare EEG to other electrophysiologic testing such as EKG and EMG, we see that the signal to noise ratio of EEG is significantly lower, which must be taken into account when interpreting the study. Now I show you this schematic of digital EEG recording just to show you all of the different places where artifacts can be introduced during the recording. So what are some common indications for EEG? We can evaluate for evidence of epilepsy or seizure risk in the form of epileptiform discharges. We can assess for focal structural or functional abnormalities, perhaps in the form of vocal slowing. We can assess the degree of encephalopathy. We can capture and characterize clinical events for differential diagnosis. If we know seizures are present, we can help to classify whether a focal or a generalized seizure disorder is present. We can use EEG to assist in the diagnosis of a specific syndrome, such as Rolandic epilepsy, Lennox-Gastaut syndrome, childhood absence epilepsy. We can capture and characterize seizures for surgical planning purposes. We can quantify how many subclinical seizures a patient is having. We can use EEG to guide the titration of medications and sedation, and we can use EEG after cardiac arrest for neurologic prognostication. However, EEG will never be used to go back in time and tell you whether a particular event was or was not a seizure. It will never rule out epilepsy and will never rule out seizures. So to get it into the types of EEG available in clinical practice, probably the most common would be your outpatient routine EEG. For primarily historical reasons, they're available usually in 20, 40, 60-minute studies, maybe more upon request. Hyperventilation and photic stimulation are usually done in an effort to provoke epileptiform discharges, and sleep deprivation can also be added but must be specified in the order, which is long thought to increase the yield of interictal epileptiform discharges. The most common indication for the outpatient routine EEG is to evaluate for the presence of epileptiform discharge, which can be important in the process of diagnosing or confirming epilepsy. The major limitations of outpatient routine EEG are it's for a finite period of time. You are unable to capture specific clinical events, and thus it can be falsely reassuring. Here's an example of some right temporal sharp waves on a spot EEG. Inpatient routine EEG, which we often call spot EEG here at UMass, is largely the same, but it's usually done either at bedside or down in the neurophysiology lab. Common indications here would be also evaluate for epileptiform discharges, but also rule out ongoing non-convulsive status epilepticus in a encephalopathic patient, perhaps with low suspicion, or to assess or approximate the degree of encephalopathy in a patient. Limitations here again, it is a finite period of time, does not rule out that the patient was having subclinical seizures, usually unable to capture and characterize clinical events, and thus it can be falsely reassuring. Here's an example of non-convulsive status epilepticus on a routine EEG. In these classic articles from Annals and Epilepsia, we see that assuming epilepsy is present, the first routine EEG will be positive for discharges in only 30 to 50% of patients, but serial EEGs in that same population show discharges in about 80 to 90% of patients. A metaphor that I like to use with patients is just because you sit by a lake with a fishing rod for 20 minutes and don't catch a fish doesn't mean that there are no fish in the pond. It might be that you weren't fishing long enough. It might be that you didn't come at the right time of day. In this metaphor, maybe you didn't capture drowsiness or sleep on the EEG. Or maybe you didn't use the right bait. In this metaphor, meaning maybe you didn't use the right provocation technique, such as sleep deprivation. To shoehorn a little bit of my own research in here, there's still significant disagreement in the literature as to whether sleep deprivation truly provokes interictal discharges in and of itself versus just increases the probability of drowsiness and sleep, during which you're more likely to see the epileptiform discharges. Just some routine EEG tips. Longer studies can increase the probability of finding discharges. Sleep deprivation can be requested, which may increase the yield of discharges. And don't be afraid to repeat an EEG if the first one was normal and you still have a high suspicion for epilepsy. In the inpatient setting, continuous EEG monitoring is becoming more and more prevalent. Ideally, the continuous record is being constantly monitored by a trained technician. We have the ability to mark and evaluate specific clinical events with a bedside button, and the length of monitoring is customized to the specific patient situation. Here, we have additional indications. Yes, we can evaluate for interictal discharges, perhaps better than with finite studies. We can assess the degree of encephalopathy. We can rule out ongoing non-convulsive status epilepticus, but now we can also capture and characterize clinical events and monitor for discrete subclinical seizures. The big limitations of continuous EEG monitoring are the video is only as good as the camera placement in the room. The quality of monitoring is dependent on the technician's ability to monitor and maintain electrode integrity. Some focal seizures may not show up on an electrographic correlate, and it's susceptible to numerous artifacts. And here's an example of a seizure that was captured on continuous EEG. In the continuous critical care EEG world, there's still no universally agreed upon terminology. Research has shown that the standardized critical care EEG terminology from ACNS does have a relatively high inter-rater reliability compared to other methods. The most updated critical care EEG terminology came out in January this year. In that terminology, specific criteria are laid out for exactly what defines an epileptiform discharges, including spikes, sharp waves, and polyspikes. This is also where we get our terminology for commonly encountered rhythmic and periodic patterns, such as generalized rhythmic delta activity, lateralized periodic discharges, generalized periodic discharges. Other modifiers can be applied to those rhythmic and periodic patterns, such as superimposed fast activity, superimposed rhythmic or quasi-rhythmic activity, and superimposed spikes or sharp waves. This is also where we get the term BIRDS, which is brief potentially ictal rhythmic discharges, which you can think of as brief rhythmic runs that didn't fully grow into seizures, but still confer the risk of seizures in the future. Not surprisingly, continuous EEG monitoring has a much higher sensitivity for seizures than serial routine EEGs. Additionally, continuous EEG monitoring is associated with favorable hospital outcomes for critically ill patients. There have been many attempts to use standardized EEG interpretation to aid in prognostication after cardiac arrest. In this 2016 paper from the Green Journal, we see that the presence of two malignant EEG features, which are defined as lack of reactivity, generalized suppression, malignant rhythmic or periodic pattern, or definite seizures, confers a poor neurological outcome. Some continuous EEG tips. It can take 40 to 60 minutes to set up a patient on continuous EEG. So if we have a high clinical suspicion for status epilepticus, including non-convulsive status, do not delay treatment in those patients while waiting for the EEG. Think about the order of operations when ordering continuous EEG along with imaging studies like MRI. You can ask us if we have MRI conditional electrodes available in those patients. Remember that there's a button, and to push the button for any events that you're hoping that we can capture and comment on. In patients who have seizures, the vast majority of seizures occur within the first 1 to 2 days of monitoring. Consider this when deciding how long to monitor. Remember, the EEG does constitute a form of tethering, which could in theory exacerbate delirium. And think, in those patients, can the patient tolerate the study, or will the leads be removed soon after placement? The epilepsy monitoring unit is comprised of hardwired beds in the hospital where we conduct elective hospital admissions, typically 5 to 10 days, where we capture and characterize typical clinical events. This can be done for differential diagnosis purposes, for pre-surgical localization and syndrome classification in patients with known epilepsy, or for the rapid titration and optimization of anti-seizure medication regimens. The primary limitations here are patients may need to take extended time periods off of work for the study, and bed availability can be a problem in the hospital, especially nowadays. It's also worth mentioning that the epilepsy monitoring unit is where we monitor patients who have undergone stereo EEG, which is stereotactic placement of multiple depth electrodes with a robot, ROSA. Now, this is for precise localization of the epileptogenic zone in patients with drug-resistant epilepsy to determine their candidacy for resective or disconnected surgeries or various neurostimulator devices. A meta-analysis in 2019 in seizure shows that stereo EEG has comparable seizure freedom rates with traditional grid and strip electrodes, but is better tolerated with fewer complications. If you have a patient who you think might benefit from an epilepsy monitoring admission, reach out to one of the epileptologists. Document in your notes each type of event you're looking to capture in detail. Let the patient know beforehand about the goals of the admission and the potential outcomes, and counsel the patient beforehand about the potential length of the stay so they can plan accordingly. Ambulatory EEG monitoring offers the same advantages as continuous EEG, but in the outpatient setting. We can often accomplish many of the same things we can do in the epilepsy monitoring unit, but notably we cannot withdraw medications to provoke seizures, and we cannot perform rapid anti-seizure medication changes in real time as we can in the epilepsy monitoring unit. But we can evaluate for epileptiform discharges, we can capture and characterize clinical events. Quantitative EEG monitoring allows us to view large swaths of time all at once. It tends to be easier for lay people to see abnormalities. The big limitations here are it does not show EEG waveform morphology, which is integral to the interpretation of most findings, and it is not a replacement for traditional EEG waveform interpretation, but it is an excellent complement to traditional EEGs. Quantitative EEG is usually available as software suites running in conjunction with the traditional EEG and can include amplitude integrated EEG, the fast Fourier transformation spectrogram, an asymmetry spectrogram, a rhythmicity spectrogram, and various proprietary automated spike and seizure detectors. One of the most interesting and famous uses of quantitative EEG trend analysis is early detection of vasospasm in critically ill subarachnoid hemorrhage patients. I guess that's it. That was an abrupt ending to keep it under 15 minutes.
Yes, thank you so much, Dr. Rossi. This was a wonderful review of the electroencephalography now that we can use at UMass, and with your colleagues and your expertise. We always find amazing what you guys can do and what analysis you can get from continuous EEG in particular. I will open the discussion if there is 1 or 2 questions. And I have to say that Dr. Chu responded, Neville, to your question in the chat. And let me see if I can find the chat. So Felicia was saying most devices were tested during long-term video EEG monitoring in admission, but I didn't see any documentation differentiating between non-epileptic versus epileptic seizure.
Okay, okay.
Thank you.
Thank you.
So my guess is right. So I have now the pleasure to invite Dr. Jadeja, who is going to complete the session on epilepsy with his talk. He's an assistant professor of neurology in our department, and I am very excited to hear his talk.
Hello, everyone, and welcome to my talk. Most of you know me already, but for those who don't, my name is Neville Jadeja, and I'm an epileptologist here at UMass. Today, I will be discussing new anti-epileptic medications. I have two disclosures to note. I do get some research support from Marinus Pharmaceuticals, as listed here, and I get royalties for my book from Cambridge University Press. I might also remark on off-label uses of medications described in the talk. So let's begin. In this talk, I will describe 4 newer anti-epileptic medications. I'm going to start with cenobamate, which is marketed as Xcopri. I'll follow this with brivaracetam, also known as Briviact. Brivaracetam is the cousin of levetiracetam, also known as Keppra, which I'm sure you are all familiar with. Finally, I'll talk about cannabidiol, which is marketed as Epidiolex, and fenfluramine, which is marketed as Fintelpa. These last two medications are most commonly used in the pediatric population and may be less familiar to adult neurologists. So we'll talk about cenobamate. Cenobamate is one of the most recent additions to our anti-epileptic drug arsenal. And it is important for adult neurologists to know about this medication, especially its adverse effects and how to use it. Cenobamate belongs to the category of sodium channel blockers. Other more well-known members of this category include phenytoin and carbamazepine. Cenobamate was approved for focal seizures, which may or may not have secondary generalization. The typical dose is 200 milligrams once daily, though you can go as high as 400 milligrams once daily in specific cases. It has been shown to be particularly useful in refractory cases of epilepsy. This was based on 2 large randomized controlled trials with a total of 655 patients. Based on the results of these trials, it was found to have at least a 55% seizure reduction compared to 24% with a placebo. This was quite remarkable, especially because 20% of patients experienced complete seizure freedom, and there was a nearly 90% reduction in secondary generalized tonic-clonic seizures. This is important because it's often the secondary generalized tonic-clonic seizures that are more debilitating than experiencing more focal seizures, such as just simple motor seizures or auras. So regarding its safety profile, there are 3 important safety features that are common to all anti-epileptic medications. These include the tendency to induce behavioral changes. The worst form is suicidality, of course, or homicidality. Withdrawal seizures from abruptly discontinuing any of these medications, and the possibility of an abuse potential, and most of the newer medications are classified as controlled substances. With regard to sodium channel blockade, the common adverse effects include drowsiness, dizziness, diplopia, disturbed gait, and problems with coordination, sometimes headache, excessive somnolence, and fatigue. 10% of patients are thought to experience this, at least based on the clinical trial data. It is important to counsel our patients, especially with cenobamate, about avoiding hazardous activities such as operating heavy machinery or driving, especially when starting this medication. In a week or 2, patients get used to this, and then they may slowly be able to resume their activities. Contraindications include known hypersensitivity to cenobamate, familial short QT syndromes. There is a tendency to alter the QT interval, and it may risk an arrhythmia. It is also contraindicated in end-stage renal disease. So the one important adverse effect that we should all be aware of with cenobamate is stress syndrome, which is a multi-organ hypersensitivity reaction that is characterized by fevers, rashes, lymphadenopathy, and multi-system involvement. This is rare, the main risk factor being rapid titration of the medication. If suspected, the drug must be discontinued immediately. Other precautions include checking the EKG at frequent intervals for QT shortening and watching for suicidal and other behavioral side effects. There are some important drug-drug interactions that we should be aware of when using cenobamate. With other anti-epileptic medications, especially phenytoin, phenobarbital, and clobazam, the doses of these medications might need to be reduced when using them with cenobamate. On the other hand, lamotrigine and carbamazepine doses might need to be increased when using concomitantly with cenobamate. I've listed there are some other drug interactions as well that I've listed here. The typical starting dose is 12.5 milligrams once daily. It is recommended that you continue this for about 2 weeks and then double the dose to 25 milligrams once daily. And then again double the dose after 2 weeks to 50 milligrams once daily, 100 milligrams after another 2 weeks, then go up to 150, and finally at about 11-12 weeks you reach the goal dose of 200 milligrams once daily. It's very important to follow this slow titration schedule, going up every 2 weeks to avoid the possibility of DRESS syndrome, as I mentioned prior. So next we'll talk about brivaracetam or Briviact. Brivaracetam is approved for focal seizures. It is available in IV form, including at UMass now in the formulary here. The mechanism of action is SV2A binding and is said to have a 15 to 30 times higher affinity to the SV2A receptor compared to Keppra. This is important because if the binding is more specific to the SV2A receptor, which is the mechanism of anticonvulsant action of these medications, then there is less of a possibility of other adverse effects such as behavioral disturbances and mood irritability, which can be seen more so in Keppra compared to Briviact. We typically start Briviact at 50 milligrams twice daily and go up in 2 or 3 weeks to 100 milligrams twice daily, which is the common maintenance dose. It is said to be more rapid acting compared to Keppra in IV form, where it quickly penetrates the blood-brain barrier, and it can be more efficacious at stopping seizures. At least that's what the marketing pitches. Now, regarding the safety profile, as I said, there are certain features that are common to almost any anti-epileptic medication, and these include behavioral changes, suicidality or homicidality being the most severe forms, the possibility of withdrawal seizures with abrupt discontinuation of the medication, and the possibility of abuse potential. Specifically, though, regarding Briviact is the issue of somnolence and dizziness and gait disturbances. There's a particular adverse effect profile which patients complain of, and the reported terminology is like you feel drunk. It is important, therefore, to counsel patients to avoid hazardous activities such as operating machinery or driving when starting this medication till they get used to it. The main contraindication is a prior hypersensitivity to Briviact or any of its ingredients, and there are rare reports of angioedema and bronchospasm. Unlike Keppra, you don't need to adjust its dose with renal impairment, but you need to adjust its dose for all stages of hepatic dysfunction. Caution has to be exercised when using this medication with certain cerebellar syndromes and with psychiatric comorbidities. Regarding drug interactions, Briviact is typically not, and the drug interactions are not typically significant. I think you have to be careful with phenytoin especially, because it can sometimes cause high phenytoin levels. Next, let us discuss cannabidiol or Epidiolex. The mechanism of action for cannabidiol, especially how it exerts its anticonvulsant effect, is still not understood. It doesn't seem to be through cannabidiol receptors. The drug is approved for 3 rare and particularly devastating pediatric epilepsy syndromes. These are Lennox-Gastaut syndrome, Dravet syndrome, and seizures with tuberous sclerosis complex. What we should know, though, is before starting cannabidiol, it is very important to get a baseline hepatic function. This should include serum transaminases, ALT, AST, and total bilirubin. And I'll sort of delve into why later. Common adverse effects of cannabidiol include somnolence, sleep disturbances, fatigue, malaise, weakness. Sometimes you may have insomnia, sometimes hypersomnolence. This is about 10% of people who use this medication. There's also a specific GI side effect profile. You can have appetite loss, nausea, vomiting, and diarrhea. There have been reports of rash, pyrexia, fevers of unknown origin, and the tendency or susceptibility to infections. There's no specific contraindication other than hypersensitivity to cannabidiol or any of its ingredients. That's rare. The main precaution that we must be aware of is closely monitoring the hepatic function. There is a risk of transaminitis. This occurs especially with higher doses of cannabidiol by itself, or if there is a concomitant use of valproic acid or another hepatotoxic agent. There is the issue of somnolence, sedation. Patients should be advised not to drive or operate heavy machinery, especially when just starting off on this drug. And it can affect behavior, sometimes causing suicidal and homicidal ideations. Regarding drug interactions of cannabidiol, there are several classes of drugs that may need dose adjustments, especially because of extensive hepatic metabolism. I've not listed all of them here, but in particular to mention is clobazam or Onfi, lamotrigine, phenytoin, and stiripentol. We don't use stiripentol that much in the US. The dose of cannabidiol may need to be increased with concomitant use of rifampin because that can induce the metabolism of cannabidiol. There is the risk of transaminitis, as I mentioned, with valproic acid. Often you have to stop one or the other, or maybe both sometimes. CNS depressants may cause severe somnolence or sedation. And as I said, that it is important to monitor liver function. Lastly, I would like to end by discussing fenfluramine or Fenterba. I'm just going to briefly describe this, not go into the details because it's indicated for pediatric patients more than adult patients. But there are a few features that neurologists should be aware of with this medication. Fenfluramine has been specifically approved for Dravet syndrome. Dravet syndrome is a very resistant and debilitating form of pediatric epilepsy. Now, it is prescribed and obtained through a risk evaluation and mitigation strategy program, also called a REMS program, and special certification is needed both by the prescriber and the dispensing pharmacy. The main adverse effects are valvular heart disease and the possibility of pulmonary artery hypertension. On the other hand, it has been shown to significantly reduce convulsive seizures in this debilitating form of epilepsy. Seizure reductions take as long as 3 or 4 weeks to set in, but they tend to remain consistent throughout treatment. Seizure reductions of as high as 65% have been reported with this medication. The main adverse effect, as I said, is cardiac function. You need regular echocardiograms before starting treatment, during treatment, and even after discontinuing the therapy. And there are some common adverse effects like decreased appetite, drowsiness, difficulty with coordination, GI disturbances. But there's one peculiar one called the salivary hypersecretion syndrome or saliva overproduction that can sometimes occur. And paradoxically, these patients might sometimes actually go into status epilepticus. It is very important to be aware that this medication cannot be used with MAO inhibitors because of the possibility of serotonin syndrome. In fact, a MAO inhibitor medication needs to be stopped at least 14 days prior to starting this one because of the risk of serotonin syndrome. So just to quickly summarize some salient features of our talk, with cenobamate, be careful to go slow because of the risk of DRESS syndrome. Check an EKG for QT abnormalities. With Briviact, remember you need to adjust the dose for hepatic dysfunction, but no doses adjustments are necessary for renal disease. It's actually a good drug to use with renal disease. For cannabidiol, be careful of the liver function, and with fenfluramine, check an echo, especially with the risk of valvular stenosis or pulmonary hypertension. It is contraindicated with MAO inhibitors due to the risk of serotonin syndrome. That's very important. Thank you, and I'm happy to take any questions that you may have.
Thank you, Neville, for this overview of the new anti-epileptics, some of them that I never heard or I knew that with different indication, like the fenfluramine. So any questions? In the meantime, if there is no question, okay, I have a question from in the chat from Dr. Chu. What are your thoughts on use IV Briviact for emergent status epilepticus?
Yeah, it has been marketed for this. I mean, I think there's an, at least from the company standpoint, they bring this up in their talks quite frequently. I think it's a good drug to use. Whether it adds any benefit from Keppra in terms of its efficacy, I don't know, because we typically don't use an anti-epileptic medication to stop seizures. For that, we use benzodiazepines. It's more to prevent ongoing seizures. I mean, whether it's better than Keppra for that is an open question. But yeah, I think it's got its place there, especially if Keppra is not a great choice for whatever reason, for mood, for renal issues, and that sort of thing.
And just a follow-up question: how pricey is this drug compared with Keppra? Like roughly, like it's the same or it's higher 10 times? Okay.
The newer ones are expensive, and obviously, like when patents expire, companies modify molecules and make newer agents. You know this better than me, but it is expensive. Most of these drugs are expensive. Cannabidiol is I don't know the Briviact cost specifically in terms of dollars. The cannabidiol is really expensive. I think it's like in the thousands.
Thank you, Neville. But the drug, you said that it's in the approval.
It's available, yes.
To be in the at UMass system, in the PNT. Okay, in the PNT. Good.
Yes. And thanks to Dr. Noviati's efforts, I think should give her credit for this. It is now available at UMass.
Thank you. And there is another question in the chat from Dr. Alam, who is asking, does cannabidiol have any off-label use in epilepsy in adults?
Yeah, so this is a good question. And these, the sort of more recent trends are to get these drugs approved through the FDA for orphan diseases or, rare sort of syndromes like cannabidiol especially is approved for Lennox-Gastaut syndrome, Dravet syndrome, and tuberous sclerosis complex. But then there is an off-label use, as I described, and we do use it. In my experience, it is no better than any other anti-epileptic in its efficacy. Just like at least in my personal experience. I think it has a certain place. And I think it becomes very important to sort of watch the liver, as I stressed in the talk, and also for drug-drug interactions. Yeah.
Thank you.
Thank you. Thank you.
I don't see any other question in the chat. I don't see any hand up. Anyone that wants to answer, to get an answer from Nabil tonight? No, if that it's, thank you, Nabil, so much for again. I want to remind everyone that Jennifer Schimmer was sending on and on, you know, the survey. You can take the advantage to do it now, or you can do it tomorrow. Hopefully, many of you will join us tomorrow at 8:30. The presentation will start at 8:40 a.m. And my special thanks goes to our supporter staff, Jennifer Schimmer and Morna Larson, that has stayed with us through a whole program, and they will even help us tomorrow in their day off. And to my both co-directors, Drs. Bosco and Dr. Silver. I wish you a wonderful evening tonight, and we'll
Dr. Ionetti?
Yep.
There is one more question.
Oh, I don't see it. Oh,
that's
good.
okay. There it is. It came up while you were talking. I just didn't want you to miss it.
Okay. Yeah. Yes. Thank you so much. Yes. Dr. Mushmagil has a question. Could any basic scientist address any potential for new targets in epilepsy? I think it's. I don't think I got any answer for you, Suzanne, but we'll keep asking this question maybe tomorrow when we will have even more, hopefully, basic researcher back with us. Again, thank you so much. Have a good evening, everyone. And I will just ask our team to stay for 5 minutes, brief feedback here and debriefing. Thank you.
Thank you.
Good night.